Hello students, and welcome to chapter 12 in our lecture 12 in our lecture series on human biology. This lecture is about the reproductive system, the human reproductive system, and more information can be found about the human reproductive system in chapter 43. Just to give you an overview of what I'm going to talk about, the first set of things I'll talk about are what are some of the other methods of reproduction that other forms of animals on Earth use. Sexual reproduction is a relatively new development on the evolutionary scale. It's something that was only developed about 300 million years ago or 400 million years ago. We believe that the Earth is about 4.5 billion years old, and there was basically no life on Earth for the first billion. So that leaves about 3.5 billion years, 3.5 billion years of life on Earth, and sexual reproduction has only existed for the last 0.5 billion or less. So it's a relatively new thing, but once sexual reproduction was introduced, human evolution and the evolution proceeded very quickly as soon as animals on Earth and life forms on Earth started to reproduce sexually. With non-sexual forms of reproduction, asexual reproduction, an organism simply splits in half and makes an identical copy of itself. And evolution proceeds very slowly if all of the organisms are the same, because the offspring look exactly like the parent, whereas if you have genetic diversity, which means that the offspring, all of the offspring that are produced through sexual reproduction, the offspring looks slightly different than the parents. And some of the offspring are different. Some of them are bigger or smaller. Some of them can endure more of this and less of that. So they're all slightly different. If there's a sudden change in the environment, whether it's a global scale change in the environment or whether it's a small change to the environment on the local scale, some of those offspring will be better able to survive within that change than others. And so they will survive, others will die out, and then those that survived had a slightly different phenotype, slightly different characteristics than their parents, and then they will in turn produce offspring that are slightly different than themselves, and those offspring will be better suited to survive if there's another local change in the environment someplace else, and so on. And so by having offspring that are different from the parents, this has created genetic diversity. Genetic diversity leads to phenotypic diversity. Phenotypic diversity means how are the genes expressed on the outside of the body? Are you tall, short, big, little? Do you have this or that characteristic? That is referred to as phenotypic diversity. And so evolution proceeds very quickly if you have phenotypic diversity, and you only have phenotypic diversity if you have sexual reproduction. Sexual reproduction essentially means that you have a bunch of genes in the genome of an organism, but every member of that species has a slightly different set of alleles. So when we were talking about the ABO blood typing system, I introduced the idea of gene alleles. A gene allele is a slightly different version of a gene. So as an example, a very simple example, within the human race, within the human gene pool, as it's called, within the human race, there are actually 3 different alleles for the proteins that are on the surface of blood cells. And we call those A, B, and O. But because humans are diploid, diploid means that every cell in the human body has 2 complete copies of the human genome. The human genome is all the genes that a human is supposed to have. And humans have about 23,000 different genes. And for each of those genes, there may be one or more alleles. There may be more than one allele. So in the case of the ABO blood grouping system, there are 3 different alleles. Each human being can only have at most 2. Many have just 1. So if you have the OO, and so your blood type O, that means you have only 1 allele present. There are 2 copies of it, but there's only 1 allele. There's only 1 gene allele. On the other hand, if you are blood type AB, that means you have 1 of each, so you have 2 alleles present. So each person can have up to 2 alleles, at least one, up to 2. And then of the 23,000 different genes in the human genome, there are about half of them have more than one allele. There is more than one type. And so what that means is that every human has a different combination of alleles. We all have the same genes, but we don't all have the same combination of alleles, because there's probably 15,000 genes in the human genome that have more than one allele. The odds of 2 people having exactly the same combination of all of those is practically nothing. So that means that every human being has different combinations of these different alleles, and that's why every human being looks slightly different. And every human being has slightly different characteristics. And some of those characteristics, if there ever happened to be a sudden change in the environment, some of those individual people would be more apt to survive in that changed environment than others. And so that would favor the survival of those individuals, and then those individuals would give rise to more diversity and so on. And that's how evolution proceeds. So the purpose of sexual reproduction, I don't know if you can say it's a purpose, but the advantage of sexual reproduction is that it leads to phenotypic diversity, which leads to evolution fairly quickly. So for 1 billion years on Earth, we just had bacteria in the ocean. Then for the second 1 billion years of life, we had bacteria plus protists. And then only in the last 300 million years have we had this explosion of different life forms that includes all the forms of plants and animals. And all of those diverse life forms usually reproduce sexually, which gives them the opportunity to change their appearance and change their phenotype, because they're passing around the different alleles and putting them together in different combinations. So the first part of the lecture, I'm just going to explain to you the different forms of sexual and asexual reproduction. So a lot of the organisms on Earth produce asexually, which means that they do not have to mate with anybody else in order to produce offspring. Those that do reproduce sexually will have different variations of what we think of as sexual reproduction. And then human sexual reproduction, I'm sure you know a lot about that already, is just one of the types of sexual reproduction that are done by, is the way that mammals do it, basically. Okay, so then we're going to discuss the details of the human reproductive system, including the male and female reproductive anatomy, the parts and how they work. Then we're going to talk about the female reproductive system in more detail, because the production of eggs is called ovulation, and ovulation is controlled by a balance of hormones. And you'll learn a lot more about how the hormones are produced in the next lecture. But for this lecture, we are going to talk about specifically how control of the production of female eggs is controlled by about 4 or 5 different hormones. It's a very complicated system. The idea of control of ovulation by hormones is that women should produce only 1 egg that is available to be fertilized every month. If human women produced the way other mammals do, if human women produce 10 or 20 eggs every month, then you would have women that can simultaneously give birth to 10 children at once. And given the construction of the human body, that's actually very dangerous. So we do have examples of human women that produce 3, 4, 5, 6, 7, 8 babies at once, but it's a very dangerous thing to do that. The mother's life is in danger every time she gives birth to more than 2. Well, frankly, the woman's life is in danger when she gives birth to 1 child, but it's more in danger when she gives birth to 2 or 3 at the same time. So given the construction of the human body, taking the construction of the human body into account, it's actually quite dangerous for a woman to give birth, to become pregnant and carry more than 1 child at a time. It becomes more dangerous the more you carry. So the hormonal control, a very careful balance of hormones, controls ovulation in human females to make sure that only 1 egg is available to be fertilized at a time. Okay, once you get pregnant, the development of the baby inside the mother's womb is referred to as gestation. And we will talk about human gestation. As you probably know, I'm sure it takes about 9 months to go from a fertilized egg to a newborn baby. Newborn baby is called a neonate, by the way, and we'll talk about that. And we'll talk about what happens when a woman gives birth. It can be a very traumatic thing, but obviously lots of women do it or we wouldn't be here. And then we'll talk about we'll also mention some common birth defects, which are birth defect. Defect is a very pejorative term now. It's a negative term, but we haven't replaced it with anything. But anyway, a defect means faulty, but there are some things that can go wrong during child development, and we will talk about the more common ones. Okay, and then because depending on where you're from and what your history is, what your background is, you may or may not have learned all about birth control as well as the control of sexually transmitted diseases. Collectively, this is referred to as sexual health. It means like you have a number of choices regarding what you can do with your own sexual equipment. And knowledge is always power. Knowledge is a good thing. And so some of you may not know all of these things, and so this is a good place to learn them. This course is a good place to learn them. So we will learn about sexually transmitted diseases, and we will learn about birth control, which is known as contraception. Contra means against, and then con means in favor of. So the word conception means to create new life. Contraception means to not create, deliberately not create life. Essentially, contraception or birth control are ways that humans have devised where they can have sex, which they enjoy, without the possibility of giving birth or becoming pregnant until they want to. So you can, in the old days, you could not separate those two things. Many people believe you still shouldn't be able to separate those two things. But nevertheless, we do have the technology to separate those two activities. That we can separate the act of sexuality from the possibility of getting pregnant. We can do that quite easily today, and there are a number of ways that it can be done. So we will learn about all of those ways. All right, let's take a look way back in history and discuss asexual reproduction. For the first 2 billion years of life on Earth, that's how reproduction was done. You had started out with 1 billion years of bacteria in the ocean. Bacteria reproduce asexually for the most part, and what they do is they simply split in half, and then you have 2 bacteria where you used to have 1 bacteria. That is referred to as binary fission. The word fission means to split in half. Okay, so asexual reproduction, let's look at this. When you have asexual reproduction, the offspring, the children if you will, the offspring look exactly like the parents. They have exactly the same phenotype. Okay, so in asexual reproduction, the offspring is an identical copy of the parent. There is no phenotypic diversity, and evolution proceeds very slowly. For the first 1 billion years of life on Earth, we had nothing but bacteria in the ocean. For the second 1 billion years, we had just bacteria and protists. And for the last 500 million years, we had everything else. All of the different, the explosion of different forms of life on Earth came about only because of sexual reproduction, which evolved about 500 million years ago, or a little bit less. So here we have a protist. This is a protozoa called a paramecium, and it reproduces by simply splitting in half, which is called binary fission. So here's the word fission over here on the right. Fission means to split in half. So we had binary fission for the first 2 billion years of life on Earth, and then in just the last 350, 400 million, we had all of this other stuff that was only made possible by sexual reproduction. All right, let's look at the 4 different types of asexual reproduction, which is possible by animals, by other life forms on Earth other than humans. Okay, so the 4 basic types of asexual reproduction are fission, also known as binary fission. Binary means 2. So binary fission specifically means splitting in half and then growing into two organisms that are identical to the original. So bacteria and many protozoa produced through simple binary fission. Budding is a slightly more advanced way where basically a piece breaks off of the adult organism and it grows into a fully grown new organism. And again, it's an identical copy of the original. So a small part grows and breaks off, and then it continues to get bigger and until it's a full-blown, full-sized organism. So the Cnidaria, if you ever take Biology 110, you'll learn that all of the life forms on Earth are grouped into something called phyla and individual phylums. For example, humans are a member of a phylum called the phylum Chordata, and that includes a lot of the animals. But there's another phylum called the Cnidaria. The Cnidaria, Cnidaria includes sea anemones and jellyfish, which of course are not fish. So we have jellyfish, sea anemones, those sorts of things, and they reproduce through budding. Okay, then there's another type of, so a little bud breaks off as if it were a plant bud, the bud of a plant. Then there's another method which is slightly more advanced, and it is called fragmentation. This is sort of like budding. The main difference is that instead of having a complete sea anemone, a little tiny miniature sea anemone break off, fragmentation means that a limb or a piece breaks off of an organism and grows into a complete adult organism. So starfish, for instance, which are technically called sea stars, the sea stars are members of a phylum called the echinoderms, which are closely related to the chordata, which humans belong to, but it's a different phylum. They reproduce through both sexual reproduction or fragmentation. So they do reproduce through fragmentation, where basically a sea star, one of the arms breaks off of a sea star, and then that arm will develop, the sea star that it broke off of will grow a new arm, and the arm that broke off will turn into a fully grown sea star. So that's called fragmentation, because you broke a fragment off of the adult organism. But sea stars can also reproduce sexually as well. So there are male and female sea stars. So they can produce sexually as well as asexually. Humans obviously cannot reproduce asexually. But there are some organisms on Earth that produce both sexually and asexually. Okay, finally, there's another interesting form of asexual reproduction called parthenogenesis. So genesis means to create or the beginning of. Parthenos is an ancient Greek word meaning virgin, referring to a woman who's never had sex before. So parthenogenesis literally translates from ancient Greek meaning virgin birth, a female that gives birth to adults without having ever been pregnant, without ever having been impregnated. So the Parthenon in Greece, which is a temple in Athens, is named after Athena, who supposedly, according to ancient Greek mythology, Athena broke out of her father's head. Her father was Zeus. One day Zeus had a terrible headache, and then bang, his head broke open, and this fully grown woman named Athena came out, and so that was a parthenogenic birth. Obviously Zeus was male, not female, so that's not a perfect analogy. But so the Parthenon in Athens is dedicated to the asexual creation of the Greek mythological goddess Athena. Anyway, in modern terms, biologists have appropriated that term to refer to insects that give birth to unfertilized, that lay unfertilized eggs that can develop into fully grown adult insects. The three best examples of that are bees, wasps, and moths. Basically, bees, wasps, and moths, they will mate, and when a male and a female bee mate, some of the eggs will be fertilized in the female and some of them will not. The eggs that, let's take a bee for instance. If a queen bee mates with a male bee, which is called a drone, some of the eggs will be fertilized and some of them will not. The eggs that were fertilized, the queen bee will lay all of these eggs, and the eggs that were fertilized will develop into worker bees, which are female. The unfertilized eggs will develop into males that are called drones, and they don't do any work at all. Their job is simply to mate with another queen when we have another queen appear. So the worker bees don't like the male drones very much, and in fact, when after a male drone has mated with a queen, the worker bees often beat up on it and kick it out of the beehive, because it doesn't do any work and it's kind of perceived to be alien. But nevertheless, a drone, now the interesting thing is that if a drone develops from an unfertilized egg, is a drone haploid or diploid? So the term diploid means that you're an organism that has 2 copies of your genome in every cell. Humans are diploid. We have 2 copies of every chromosome. The complete human genome is made up of 23,000 genes that is spread out over 23 different chromosomes, and we have 2 copies of each of those chromosomes. That means therefore that humans are diploid. Female worker bees arise from fertilized eggs, and those eggs, the eggs and the sperm, are both haploid, meaning that they have only 1 copy of all the chromosomes and therefore only 1 copy of all the genes. So an egg, before it's fertilized, both a human egg and a bee egg, before it's fertilized, they are both haploid. Once the egg is fertilized with a sperm, then we have the set of chromosomes that was already present in the egg being combined with the set of chromosomes that was in the sperm, and therefore you have a diploid fertilized egg that develops into a fully adult diploid adult. Okay, what about these bees? So some of the eggs are fertilized, which means they develop into diploid workers. So the worker bees in a beehive are all female and they are all diploid. The male bees, whose only job it is to mate with a queen, are haploid. So that means they only carry one copy of each chromosome. So parthenogenesis refers to the development in bees, wasps, and moths where unfertilized eggs are capable of developing into fully grown adult insects that are haploid. Okay, so here's another protozoa that is, this one is called Giardia lamblia, by the way. Lives in the water. It reproduces through binary fission, so you get 2 organisms. It just simply splits in half, and then you have 2 identical organisms. Now we move on to the budding. So here we have a sea anemone, a sea anemone, one of those things that normally grows on the ocean attached to a rock. And you see that it has some little buds that grow off, and then eventually those buds will break off and then expand in size, and they'll become a new sea anemone. That's budding. So here we see a coral reef with some little buds appearing off. The little buds have actually broken off and attached to a rock all by themselves. Okay, so those Cnidaria, the sea anemones and the jellyfish, sea anemones in particular, are the main things that populate a coral reef, by the way. So here we have a beautiful coral reef that is populated by different forms of Cnidaria, including sea anemones. And then usually the coral is the skeleton, the skeleton of a dead sea anemone. All right, here we have a sea star. One of the legs gets big and then breaks off, and then the leg that broke off is replaced because the sea star develops a new one. Sea stars normally have 5 legs, but there are other types of sea stars that have more than 5. So here we see a leg has broken off one of these sea stars, and it has grown a new leg, so it has 5. And then the leg that broke off is in the process of sprouting 3 more little legs, and so eventually it will have 5 as well. So that's an example of fragmentation. That's an example of fragmentation. Here we have some bees. The bee on the far right, the largest one, is the queen bee. Over on the far left we have a worker bee, and then we have a drone bee in the middle. So the queen and the worker are both diploid, and the drone developed from an unfertilized egg. Right? So that is called parthenogenesis. The queen is diploid, the worker bees are diploid, so we do not consider them to be examples of parthenogenesis. But the drone, the male drone, is haploid, and he developed from an unfertilized egg, and so that is what we call an example of parthenogenesis. Okay, so summary of the asexual forms of reproduction. I could ask you about these on a test, right? So we have binary fission, which is the way bacteria and protozoa usually reproduce. We have budding, which is the way hydras and members of the class Cnidaria, the phylum Cnidaria rather, reproduce. We have fragmentation, which is the way sea stars reproduce. And remember that sea stars are also capable of sexual reproduction, so they don't have to reproduce by fragmentation. And then we have parthenogenesis, and remember that insects like bees, wasps, and moths can also reproduce sexually. So the queen bee gives birth to both fertilized and unfertilized eggs. Both the fertilized and the unfertilized eggs develop into adults. It's just that the males are haploid, so only the males. Are an example of parthenogenesis. And bees, wasps, and moths generally are practicing both sexual and asexual reproduction at the same time. Most plants, in case you didn't know, most of the more advanced plants are what are called hermaphrodites. A hermaphrodite is a plant that has both male and female sexual organs. So their plants are not reproducing asexually. Most of them are not. A few of them do. Okay, so the offspring are identical to the parents. This is not the best idea because we have very little phenotypic diversity. Very little phenotypic diversity happens when you have offspring that are identical to the parent. So they are unable to respond to changes in the environment. For example, the ocean is heating up due to global warming. Anyone who tells you that global warming is not certain is either lying to you or they're being taught by somebody who's lying. And so the planet is heating up. Yes, the winters are still cold. Yes, we still have unusually cold winters now and then, but on average the planet is heating up. The ocean is warming by about 1 degree the ocean is warming by 1 or 2 degrees at this point. Now you may say, what's the big deal, 1 or 2 degrees? Well, the big deal is that most of the Cnidaria cannot tolerate a change in temperature. So humans are very hardy animals. We can tolerate large changes in temperature, but not every animal is tolerant to temperature changes as humans are. A good example are the Cnidaria. So the Cnidaria that make up the sea stars, the sea anemones, and all of the coral reefs, for instance, they cannot stand a change in even 1 degree. And so the coral reefs around the world are dying because the ocean is now 1 or 2 degrees hotter than it used to be. So we're losing all of our coral reefs, right? And then the other thing is that the algae in the ocean, you know that we get our oxygen from plants. You're familiar with land plants, but where we get most of our oxygen is actually from the algae in the ocean. The algae in the ocean are photosynthetic protists, and so they take light from the sun and they turn it into oxygen, which we breathe. And ocean algae are not very tolerant to changes in temperature either. So that means that the worst possible scenario for global warming is that we lose the oxygen that's being produced by the ocean because the ocean warms up, right? So it's not just a matter of being hot in the summertime. We are getting hotter and hotter summers, but that's not the scary thing about global warming. The most terrifying possibility about global warming is that if the algae start dying off, then we have no more oxygen to breathe. So that is the actual threat that's being posed by global warming, and that's why it's imperative that we do something about it. Anyway, so there are many organisms that can't stand a change in temperature, and so the Cnidaria, the sea anemone and the coral reefs are examples of organisms that reproduce asexually, and they're all the same, and all of them are the same in that they cannot withstand a change in temperature. There are other organisms, obviously, that are diverse enough that some of them can. Some of them can't, some of them can. So there's some humans that can withstand a change in temperature better than others, either up or down. Okay, so anyway, evolution proceeds very slowly, and it's also for organisms that reproduce asexually, they are very vulnerable to becoming extinct if something happens to the environment that they can't tolerate. There's some change in the environment that they can't tolerate, they will all die, as opposed to some of them surviving, which is what you have with phenotypic diversity. All right, now let's look at sexual reproduction. So remember I said that there are different alleles. Alleles are different, slightly different versions of a gene. Humans are diploid, so we have two copies of each gene, and we may have, those two copies may be the same, in which case we have one allele, or they may be different, in which case we have two alleles. If you take Biology 110, you'll learn that when you're talking about one particular gene, if a person or an animal has two copies of the same allele, there's only one allele present, we call them a homozygote. And if a person or an animal has two different alleles, we call them a heterozygote. And so if you take Biology 110, you'll learn all about that. It's part of genetics. Genetics is a very fascinating field of study. Okay, so we have multiple alleles in the population. Humans can be either a homozygote or a heterozygote, meaning that they have either one allele or two alleles present, regardless of the fact that we have two copies. If there's only one allele, two copies of the same allele, we say that there's only one allele present, and that person is a homozygote. Okay, so we have different alleles for different genes that are being passed around between individuals, right? So we have, we have males and females in the human race, for instance. There are other organisms that don't have males and females, but they still reproduce sexually. And so these individuals that have different combinations of alleles are reproducing with each other to create even more combinations, even more different combinations, and that's the idea. So there are different gene alleles that are being passed around within a population of organisms. So as I said, there's 3 different copies of the ABO blood group alleles. Each human being has 1 or 2 at the most, but there are 3 in the population at large, and so we're passing them around to each other in different combinations. So this results in phenotypic diversity. Phenotypic diversity means that a species can survive a change in the environment if not all of the organisms are the same. You have some diversity, and that's, you can talk this is a philosophical viewpoint of mine, which is that, and you can talk about it with the sociologists and see if they agree. But Canada and many other countries have a diverse population, which means that we have all kinds of different people who originally came from different parts of the world, and they have different viewpoints and they have different value systems, and some of them are better at certain things than others. Suddenly, if you need people who have a certain attitude or a certain set of skills, you've got them. Whereas there are other countries that are deliberately homogeneous. Homogeneous meaning all the same, and they don't tolerate diversity very well. And so suddenly, if the environment or the situation of that country changes, and you need people who have a different skill set or different attitudes or different behaviors, you don't have them. You may or may not have them. So the entire society may suffer. Whereas in diverse societies, the society can kind of turn to the people who have the skills or the attitudes that you need in order to survive a change in the situation. So for that reason, obviously, I think that diversity of a society is a good thing. And so Canada is a fairly diverse society. We need to work on being even more diverse and even more accepting of different people and different attitudes, as every country should, I think, in my opinion. But that's a philosophical question. That has nothing to do with biology. Biologically speaking, if you have phenotypic diversity, different animals of the same species that are different and have slightly different characteristics than other members of the same species, this can lead to natural selection and surviving changes in the environment. Okay, so as I said, we had 2 billion years where the world was quite boring, and the organisms that inhabited the Earth reproduced through binary fission, and then in just the last 350 million years, we've had this explosion of diverse forms of life, which were facilitated by sexual reproduction. Okay, so 2 billion years boring, 300 million years, very exciting. So now, to give you an illustration of how different combinations of alleles can give rise to phenotypic diversity, I want to show you the example of dog breeding. Okay, so I had a friend once who was trying to tell me, he was trying to lecture me about how the theory of evolution can't possibly be true, because he was saying that, you know, if you went, how did some horses evolve into giraffes? Right, so there are some parts of the world where the leaves on the trees there are leaves on the trees up above the ground. A giraffe has an advantage because it can eat the leaves off the trees even though the trees are tall. So if the theory being that you had some sort of an animal that looked like a horse, and it evolved a long neck. And so my friend was saying, but you know, that's ridiculous. I mean, if you're waiting for a gene mutation that causes a long neck, what about all the other mutations that wouldn't cause a long neck? I mean, can you imagine all the freaky, unusual animals that have a long tail or a long ears or something like that, that don't give you the advantage of, you know, how many of those genetic freaks would you have to go through before you eventually got to one that had a long neck? And so the truth is, you know, I explained to him that, you know, that's not what evolutionary people believe. That's not what evolution and natural selection is. What evolution and natural selection is the idea that there are different gene alleles, and those different gene alleles arose through mutations, and then those different gene alleles are passed around between members of the same species in order to give you differences in phenotype, differences in appearance and characteristics, and then some of those differences in characteristics are better able to survive a change in the environment than others, which leads to evolution into a new species. So we didn't have to wait around for horses to sustain mutations one at a time before we ended up with a horse with a long neck. In fact, horses with longer necks were probably happened as a result of putting together different combinations of gene alleles without ever once creating a new allele through the process of mutation. So new alleles are created through the process of mutation, but that's not really what drives evolution. What drives evolution is putting those new alleles together in different combinations within the same species. So the best way I can think of to show you how that works is by dog breeding. So let's take the example of dog breeding. These are all the different strains of dogs that we've created. These are all the same species, but they all have a very different appearance because people selectively bred. They put together, they put together different dogs, and then they had funny-looking pups, and they said, well, those look funny. Why don't we mate those puppies together and see what their puppies look like? And then we keep on making sure that we're only breeding these funny-looking dogs with other funny-looking dogs that have the same appearance until we get what's known as a purebred line. So these are all the different purebred lines of dogs. But these dogs, not one of these dogs was created by a mutation. Every one of these different funny-looking dogs was created by putting together different gene alleles that already existed in the population of wolves. So, you know, wolves contain the population of wolves as a whole. The general population of wolves contains all of the gene alleles, many different gene alleles for every different gene. It's just that these gene alleles are rarely seen. The mutant or the diverse gene alleles are rarely seen if wolves are allowed to mate with each other without any preference. If wolves are allowed to mate with each other randomly, you will rarely see a funny-looking wolf get born. But as soon as humans started domesticating wolves, they noticed, oh, there's every now and then a funny-looking puppy would come out of a wolf, and then they'd say, well, let's see what happens. There's two of them that came out that look funny. Let's see what happens if we mate those two funny-looking pups together, and then they produce even funnier-looking ones. And then if we make sure that we don't allow them to mate with a regular wolf at any point, we can create a purebred dog that has a very different appearance from the original wolf. Okay, so we went from this, the wolf, all the way to this without making a single new gene mutation. That is quite a drastic change in phenotype, which is simply the result of putting together different alleles of genes. Right? So there was never one new. We went from the wolf to this little toy dog without ever once creating a new mutation. It was simply a matter of putting together different combinations of the different gene alleles. So the same thing can happen with other animals as well. And so humans managed to go from the wolf on the left to the toy dogs and all the other dogs on the right in a mere 5,000 years. It's probably 5,000 years or so that humans have been selectively breeding dogs into different breeds. So the idea is that if humans can go from the wolf on the left to the poodle on the right in a mere 5,000 years through selective breeding, deliberate selective breeding, the same sort of thing can happen by accident, except that it will take a much longer period of time to happen by accident. And of course, we have lots of time. If sexual reproduction has been around for 3.5 million years, and we managed to convert wolves into poodles in 5,000 years simply by deliberate selective breeding, surely you can have something similar like that happen to all sorts of animals by accident, by selective breeding that happens by accident, often by geographic isolation. So you have the wolf population. Just imagine this scenario: you have the wolf population that are living on a landscape, some area, and then there's a volcanic eruption or a landslide that cuts off part of that wolf population from the main body of the wolves, and then you have like half a dozen wolves that are now isolated from the other wolves, and they start breeding with each other, and then you start seeing these not mutations, but you start seeing this unique combination of alleles start to appear. And that is in fact how we believe most of the evolution took place. You had one type of animal that got split into two groups and isolated from each other, and then they got split into a smaller group, and when the smaller groups start breeding with each other, you get a totally different set of phenotypes, and eventually those phenotypes, if you try to put them back together again, they will not breed with each other if you put them back together again. And if you take Biology 110, you'll see that is something called speciation. It's where you have the evolution of new species, which results from taking an old species, splitting it in two, usually through geographical isolation, and then you split them into two small groups, and then those small groups begin to take on characteristics that are unusual because they were drawn from a smaller gene pool, and then you end up getting these unusual. Eventually, if they're separated long enough, they will no longer resemble each other, and they won't breed together, and eventually they'll lose the ability to breed with each other, and that's called speciation. So speciation is the creation of new species as a result of geographical isolation or other forms of isolation. Okay, so natural selection is basically the natural version of selective dog breeding. So instead of humans deliberately mating together these two funny looking puppies, the environment does this by accident and then ends up creating some environment that selects for a type of dog or a type of animal that is better able to survive in a changed environment. So natural selection, that is called natural selection when you have phenotypic diversity, and then one of the different phenotypes is more capable of surviving in a changed environment than others. Okay, so that's called natural selection as opposed to deliberate breeding, which humans did with dogs. Okay, so let's look at some examples of natural selection. We believe that all of these different forms of life on Earth arose through natural selection over a period of billions of years, but that's a theory, but we've seen it happening just in the last 200 years that we've been watching. So the theory of evolution and natural selection is only 250 years old, and so we've been watching to see if this happens. And just in the very brief period of 100 years, 200 years that we've been watching, we have seen examples of natural selection take place, and I'm going to give you a couple of examples of that. Okay, so the first example I want to share with you is the peppermoth population. There's a type of moth called the peppermoth, which was present in England during the Industrial Revolution. The Industrial Revolution in England caused a dramatic change to the environment, and that resulted in a dramatic natural selection of certain phenotypes of pepper moths, and I'll tell you about that. And then I'm going to tell you about the evolution or the natural selection of finches, a certain population of finches on certain of the Galapagos Islands that happened after brush fires, forest fires. So let's start with the pepper moths. Okay, so in England we have a bunch of country gentlemen who practice the science of lepidopterism. Moths, the technical term for a moth is a lepidopteran. There have been, for hundreds of years, there have been people in England that are interested in collecting butterflies and moths as a hobby. So for a few hundred years, we have data going back, information about the phenotypes of moths in England that goes back a few hundred years. So before the Industrial Revolution, the pepper moth is what is called dimorphic. Di meaning two and morphic meaning forms or appearances. So the dimorphic forms, before the Industrial Revolution, about 99% of the pepper moths were white with black spots, and 1% of the pepper moths were black. Right. Now, the pepper moths lived in parts of England that were populated by birch trees, and so the phenotype where they were white with black spots gave them perfect camouflage to be sitting on a birch tree and be invisible so that birds can't eat you. So these pepper moths were in danger of being eaten by birds, provided the birds could see them. Okay, can you see the pepper moth sitting on that birch tree there? The top one is very difficult to see because it blends in perfectly with the birch tree. It has camouflage, right? Now the black one stands out like a sore thumb, to use that expression. It sticks out like a sore thumb. It's very obvious. And so the birds can see it very easily and they eat it very easily, which explains the reason why before the Industrial Revolution, 99% of the pepper moths were white with black spots and only 1% were black, because the black ones were very obvious to see, easily spotted by the birds and easily eaten by the birds. Okay, then Industrial Revolution, 1860 or thereabouts, they built all of these factories in northern England that were spewing out coal dust and soot. The coal dust and soot got stuck to all the sides of the birch trees, and the birch trees went from being white with black spots to being black. Industrial Revolution. Suddenly the black one is invisible and the white one is very obvious and easily eaten by birds. And in a period of about 10 years, the peppermoth population went from being 99% white and 1% black to being 99% black and 1% white. It went through a period where the population went way down as the birds were picking off and eating the white moths, but that left the black moths alone and they mated with each other and they recovered. So the area was repopulated by black moths. And so now for a long time the pepper moth population in northern England was 99% black and 1% white. And now they're starting to shut down some of those awful factories, and so they don't pollute so much, and the birch trees are going from black back to being white again, and so the moth population is going back. Now that's a perfect example of natural selection. There was a change in the environment, which the pepper moths have a phenotypic diversity. They come in either black or white colors. The trees were white. There was a sudden change in the environment that led to a sudden change in which phenotype of the pepper moth was able to better survive in the changed environment. So that's a perfect example of natural selection that happened just during the short period of time that we were watching to see if natural selection was a real thing. Next example, The Galapagos Islands are a group of islands where Charles Darwin first started exploring and came up with the idea of evolution and natural selection. In reverence to Charles Darwin, the world decided to make a law that humans could not modify those islands. Those islands are basically a natural reserve now, where nobody but scientists are allowed to go and watch evolution in progress. So these islands were set aside, the Galapagos Islands were set aside in the early 20th century and reserved for scientists to just watch what's going on and with evolution. Now, on these different islands, you have different populations of finches, these little birds that you see on the right, these little finches. Now, finches have, if you've ever seen finches, you know that there's some finches that have smaller beaks and some finches that have larger beaks. Most of the finches have a beak that's about this big, but there are a few finches that have a bigger beak. Now, that is an example of phenotypic diversity. The average finch has a beak this size, but a few of them have larger beaks and a few of them have smaller beaks. That is phenotypic diversity. Now, the average sized finch, the average finch has a beak that is just the perfect size to break open and eat grass seeds. And that's what most of the finches on these islands eat. There are a few other finches that have a bigger beak, which is not ideal for breaking open grass seeds, but they can break open tree seeds. They can break open the bushes on the seeds on bushes. All right, now on one of these Galapagos Islands, they had a brush fire, right, and all of the grass burned away, which meant that all of the finches with regular-sized beaks starved. And those finches that were eating the seeds off of the bushes instead, they were the only ones that survived. So the finches with the average size beak disappeared overnight in the period of just a few years, and the finches, so the general population of finches declined drastically. But most of the ones that survived, you know, the numbers are much smaller now, but the ones that did manage to survive had a bigger beak, and they started mating with each other. And the beaks, they basically ended up becoming the new average beak size. So now we have an island that's populated by finches that have a larger beak. And so right there, again, just in the short period of time that we were watching to see natural selection in progress, we saw that. And there are about a dozen examples of natural selection that scientists have observed just in the last century. And so we think, well, you know, if that kind of thing is, if you can have a dozen good examples of natural selection happening in just the last 100 years, just imagine how many times it happened in the last 300 million years. So this, by the way, is a graph that shows the average beak size for the finches on that particular island before the fire. Right? So the average finch beak was about 8 millimeters, 8.5 millimeters, and then after the fire, a mere 2 years after the fire, it was a drought that gave rise to a fire, by the way. And so after the drought and the fire, the average size of beaks on the finches was about 9.5. So it went from 8.5 to 9.5 millimeters just in a couple of years as a result of a change in the environment. Okay, so to summarize, sexual reproduction is an advantage because it leads to phenotypic diversity. Phenotypic diversity is brought about by the fact that you have several alleles of each gene that are being passed around in the population. The different alleles were originally caused by different mutations, but evolution is not really driven by mutation. It's driven by having mutations that can then be mutant alleles that can then be put together in different combinations, leading to phenotypic variation. So the advantage of sexual reproduction is that evolution proceeds very quickly. All right, so gametogenesis. One thing that you need to, before we can proceed to talking about reproduction, we need to talk about gametogenesis. So briefly, eggs and sperms are haploid. Humans are diploid. The human egg is haploid. The human sperm is diploid. When you have diploid sexual reproduction cells, namely eggs and sperms, they are referred to as gametes. Some of you may be familiar with game theory. That's, what happens when you put different things together in different combinations. Each one of those things that can be put together in a different combination is called a gamete, right? So how are our gametes created? That is called gametogenesis. So if all of the cells in the human body have 2 copies of each chromosome, how do we go about getting these cells that have only 1 copy of each chromosome? How do we go about getting these cells that have only 1 copy of the genome in each cell? And that is called gametogenesis. So sperms and eggs are gametes. How are they created? Well, they're created through a process called meiosis, which we'll talk about. Okay, so we already know about haploid versus diploid, and we know that diploid organisms create gametes. These are eggs and sperms in humans in order to create new combinations of gene alleles. So you put together a haploid egg and a haploid sperm that have different combinations of genes, and then you get a new offspring that has a totally different combination of genes than either its mother or its father. Okay, so you should know, I will test you on this, you should know that regular cell division, when a regular cell that has 2 copies of each chromosome divides in half, that is, and creates 2 daughter cells, when a cell divides in half, it creates 2 new cells that are referred to as daughter cells, not sons, not nephews, daughter cells. Right? So when a regular cell, which is called a somatic cell, when a somatic cell divides in half, that process is called mitosis. Mitosis, you start out with a parent cell that has 2 copies of each chromosome, and then it divides in half so that you have 2 daughter cells, each of which also has 2 of each chromosome. So in order to do that, before the cell divides, it had to make 2 more copies of each chromosome. Right? So a regular somatic cell has 2 copies of each chromosome. Just before it's getting ready to divide, it copies each of those chromosomes so that it has 4. Right? So it has, just before a somatic cell divides, it goes from having 2 copies of each chromosome to having 4 copies. It's a temporary tetraploid. And then the cell divides in half, and each of the 2 daughter cells has 2 copies of each chromosome. Right? So when that happens, that's called mitosis. Now, meiosis is like mitosis in that you start out with a cell that's about to divide. It has 2 copies of each chromosome. It copies each of those 2 copies to make 4. So you have a cell that is about to divide in 2. It copies the chromosomes so that it's got 4 of each, and then it divides in 2. So you have 2 cells that have 2 sets of each chromosome. But those 2 daughter cells don't stop there. They divide a second time, except the second time they divide, they do not copy the chromosomes first. So after the second division, you end up with 4 cells, each of which only has 1 copy of each chromosome. And that cell is called a gamete. Right, so when you have 2 rounds of cell division, and the second round of cell division is not preceded by copying the chromosomes, that is referred to as meiosis. And so we call the first round of cell divisions, we call that meiosis 1, and the second round we call meiosis 2. So that means that during the process of meiosis, 2 rounds of cell division, during meiosis 1, the cell copies the chromosomes and divides, so you end up with 2 cells that have 2 sets of chromosomes. Those 2 cells then divide again, so at the end, the result of meiosis 2 is that you have 4 cells, each of which has only 1 copy of the genome, each of which has only 1 copy of each chromosome. Right, so in a regular somatic cell, you have 46 chromosomes. Just before division, you divide each of those chromosomes, so you have 92 or whatever it is. I'm not very good at arithmetic off the top of my head. So you copy them first, and then the cell divides. Now you have 2 daughter cells that each have 46 chromosomes, and then each of those cells divides a second time, so that you now have 4 cells, each of which has 23 chromosomes. and contains one complete copy of the genome that either came from the mother or the father. If it came from the mother, you're talking about an egg, obviously. If you came from the father, you're talking about a sperm. Okay, so regular cell division is called mitosis. The type of cell division that leads to gametogenesis is called meiosis. So I will probably ask you about that on a test. Okay, so mitosis results in diploid cells that has two sets of the chromosomes, two of each chromosome, 46 chromosomes in total. One of each of chromosome 1, two of each of chromosome 1, two of each of chromosome 2, two of each of chromosome 3, and so on. We literally number the chromosomes, chromosome 1, chromosome 2, chromosome 3. Okay, during meiosis, you do another round of cell division right afterwards, and you end up with 4 haploid eggs that has one set of chromosomes, one set consisting of 23 chromosomes. Okay, now in mammals we have permanent sexes. We have male and female. Some of you may realize that there are other life forms on Earth that do sexual reproduction without having two sexes. The worms, for instance, the worms have their own phylum, which is called the phylum Annelida, which you'll learn about if you take Biology 110. The Annelida are generally hermaphrodites, which means that worms have both male and female sex organs. They produce both eggs and sperms, and they are in fact capable of mating with themselves and producing offspring by putting their own sperms onto their own eggs and fertilizing their own eggs. But they can also reproduce. They can also achieve more diversity if they mate with another worm as opposed to mating with themselves. Okay, so the hermaphrodites, which are mostly members of the phylum Annelida, they're hermaphrodites, and they're capable of sexual reproduction with themselves, but you don't actually get much phenotypic diversity if you're mating with yourself. You get more phenotypic diversity if you mate with another worm. Now, there are some organisms that reproduce sexually that do not have a permanent male and female, but mammals have a permanent male and female. So in male mammals, the creation of the gametes are called spermatozoa. So in male gametogenesis, the gametes are referred to specifically as spermatozoa. And the creation of spermatozoa is called spermatogenesis. The word zoa means motile, by the way, and the word motile is a fancy word for mobile. Okay, so then the female production of gametes is referred to as oogenesis, and the egg that results at the end is called an ovum, plural ova. Right? So the technical name for a female egg is an ovum. The technical name for a male sperm is a spermatozoa. The creation of spermatozoa in males is called spermatogenesis. The creation of ova in females is called oogenesis. Okay, so ovulation, ovu contains the word ovum, root word ovum. Ovulation is the monthly production of a mature egg, one egg available to be fertilized every month. If that egg is fertilized and the woman gets pregnant, the hormones will change what they're doing and prevent all of the other eggs from maturing until that child is born. Right, so a regular human woman produces one egg that's available to be fertilized every month. If that egg is fertilized, the hormones make sure that her ovaries stop producing eggs until after that baby is born. So there's no more ovulation for 9 months until the baby is born, and then you can resume ovulating as usual. All right, fertilization. You have an ovum and a spermatozoa that get together. I'll show you the structure of a spermatozoa later, but basically only the head of the sperm goes into the egg. So the female keeps hold of the egg. The male sperm penetrates the egg and fertilizes it. And a fertilized ovum is renamed a zygote. Right, so I will test you on the fact that an ovum is an unfertilized egg. A fertilized egg that has been fertilized by a sperm is called a zygote. So a zygote is a single cell that now contains the chromosomes that came in with the sperm and the chromosomes that were originally there from the from the mother. So the father's chromosomes have now been put into the nucleus of the unfertilized egg, the ovum, and now we have a diploid egg. That egg will divide a few times, several times. The first few times that cell divides, the cells will be identical, and so they're kind of generic cells, you know, a general unspecialized type of cell that looks kind of like a soccer ball, and that is called a blastula. The blastula eventually turns into an embryo, and within an embryo you have a little miniature developing person that has organs, and very, very specialized tissues, very specialized cells. So the process of going from a blastula where all the cells are the same to an embryo where the cells are different is called embryogenesis. And organogenesis takes place during that period, which is typically the first 2 months. The first 2 months of development, you have organogenesis taking place, which means the organs are developing. Okay, then you have, once the embryo has developed and looks like a small human, has all the proper human organs, are finished being produced, it's called a fetus, but it hasn't come out yet. It looks like a small human being that just hasn't been born yet. The fetus will then, during the last couple of months, it will develop a thick layer of fat tissue, of adipose tissue. So babies, you know, they come out being very fat, and which is normal. If a baby is born prematurely, you notice that, like if they're born after 5 months or 6 months or 7 months, they look very wrinkly because the adipose tissue has not been put underneath the skin yet. So when a baby comes out, they don't have wrinkly skin because the skin has been filled out by all of this adipose tissue. Okay, so but then the fetus fattens up, and then when it comes out, it's referred to, after birth, it's referred to as a neonate, which is why the hospital ward where all the newborn babies are is called the neonatal ward, or the neonatal clinic. So here we have a bunch of sperms that are attacking a single egg. One of them will penetrate the plasma membrane of the egg and get inside. The penetration of the egg by one sperm will cause a chemical change in the plasma membrane, which prevents any of the other sperms from getting inside. That's a mechanism to prevent an egg from being fertilized by 2 sperms at once, because then you would have 3 genomes in there, which would lead to mutations. That would lead to a lot of birth defects if you had a triploid zygote. So you want a diploid zygote, but not a triploid zygote. Now, the fact that the egg came from the mother and the only part of the father that got into the egg was the head of the sperm, that means that all of the organelles that are present in the egg came from the mother. So that includes the rough endoplasmic reticulum, you know, whatever else, but significantly, it means that the mitochondria in the cell all came from the mother. Now, mitochondria were originally independent living bacteria that have their own chromosome. So bacteria, in case you didn't know, bacteria generally have one chromosome, and it is circular. Mitochondria that are inside human cells were originally independent bacteria that entered a protist a billion or so years ago. And so that means that everyone's mitochondria has its own chromosome, which is separate from the other genes that are located in the nucleus, right? So every mitochondria has its own genes, and that is what we often mean when we refer to as maternal inheritance. The word maternal means mother, and inheritance in this case means the genes that you inherited. Now, what that means is that because it was the mother's egg who was fertilized, the father did not contribute any mitochondria. So the mitochondria that I have are identical to the mitochondria that my mother has, which are identical to the mitochondria that her mother has, that are identical to the mitochondria that her mother has. So you can trace somebody's maternal ancestry, meaning you can figure out definitively who somebody's mother is, by looking at the genes, by doing a genetic fingerprint of the mitochondrial DNA. So you look at the genes that are in a person's mitochondria, and they will be identical to the genes that are in their mother's mitochondria. So geneticists are interested in this because you can determine somebody's maternal lineage this way. So let's say that you have a child and you're not sure who the mother is. You can prove it definitively by comparing the child's mitochondrial DNA to the mother's mitochondrial DNA. That will not tell you who the father is, but it will tell you who the mother is, and it will tell you who the mother's mother is, and it will tell you who the mother's mother's mother is, and you can trace somebody's maternal lineage. You can trace somebody's mother, maternal grandmother, maternal great-grandmother, and so on. You can trace their maternal lineage exactly by looking definitively by looking at the mitochondrial DNA alone. Right? So, for example, just as an example of that, you may remember that Russia, the country Russia, used to have a czar who was like a king, right, before the Russian Revolution. And then in the early 20th century, there was a communist revolution in Russia where the communists took the Tsar Nicholas, and his wife Alexandria, and all of their children. They deposed them, and to make sure that they couldn't get back on the throne, they murdered them all. Basically, they took them out into the woods and machine gunned them all to death, buried the bodies in an unmarked grave so that nobody would ever find them, and nobody could ever, say, oh, that's the body of the Tsar. Okay, so in the 1990s, somebody dug up some bodies in a forest in Russia and said, I wonder if that's, there's exactly the right number of people to be the Tsar's family. Is there any way we can tell? And the answer is yes, because Prince Philip, who just died, the husband of Queen Elizabeth, Prince Philip is, and the Tsar had the same maternal grandmother. So they took some cells out of Prince Philip, and they looked at the mitochondrial DNA, and they compared the mitochondrial DNA from Prince Philip to the mitochondrial DNA that they found in these skeletons, and they found that yes, that those are indeed the skeletons from Tsar Nicholas and his family. And it was because of maternal mitochondrial DNA, the fact that it doesn't change, it doesn't get rearranged the way other alleles do. So all of the other alleles that you get when you fertilize an egg, they will all be scrambled into a different combination. The one exception, there are two exceptions to that. One of them is the mitochondria, which you inherit exclusively from your mother. So you don't inherit a different combination of mitochondrial alleles as a result of having two parents. That's one thing that never changes. The other thing that never changes is the Y chromosome. So the Y chromosome is only given to boys, and the genes in the Y chromosome are identical to the genes in the father's Y chromosome and his father's Y chromosome, and so on. So if you're trying to determine without doubt, you're trying to determine definitively without doubt that who a boy's father is, you compare the gene alleles present in the Y chromosome, because the father donates the Y chromosome directly to the son. So you can determine without doubt, you can determine unambiguously who a boy's father is, who the father's father is, who the father's father's father is, and so on. So you can determine you can determine without doubt who somebody's mother is, who somebody's mother's mother is, who somebody's mother's mother's mother is, and if you're talking about a boy, male, you can determine who his father is, who his father's father is, who his father's father's father is, because the Y chromosome is passed down from generation to generation unaltered, because it has no chance to recombine with any other combination of alleles. So anyway, the fact that you inherit your organelles in your cells exclusively from your mother is significant. And if you take Biology 234, you'll learn a bit more about how that can be used and why that is significant. By the way, geneticists call it maternal effects. There are a lot of things that happen with your genes when you're developing in the beginning that are the result of having inherited the egg from your mother. Okay, so there are the mitochondria. The mitochondria in the cells, everybody's cells, came exclusively from their mother. The mitochondria have their own chromosome, which is not present in the nucleus. And so you can, by looking at the mitochondrial DNA, as they call it, by looking at the mitochondrial DNA, you can determine unambiguously who somebody's you can determine somebody's lineage, somebody's maternal lineage, unambiguously. All right, so we fertilize an egg. We start out with a zygote. It used to be an ovum. As soon as the father's chromosomes get into the nucleus, we call it a zygote. Then the cell will divide a few times and eventually form what's called a blastula, a hollow ball. It's hollow on the inside. Then one side of it will pinch inward to form the digestive tract, and then that process is called gastrulation. I could ask you about that on a test. And once you've got this part formed, this is called a gastrula. Now notice that when we had a soccer ball, we didn't really have an inner layer of tissue versus an outer layer of tissue. But as soon as we go to a gastrula, you have an inner layer of epithelial cells and an outer layer of epithelial cells, and then an in-between layer, right? So the outer layer, the inner layer, and the in-between layer are going to develop into 3 different sets of tissues and organs in the adult body. So this is significant. The study of looking at which parts of a gastrula turn into which parts of an adult is called embryology, and it is a branch of biology as well. So there are scientists who spend their whole life studying the development of embryos into adult organisms. All right, then we develop from a gastrula into an embryo, so more embryology, which looks like a little tiny tadpole. A tadpole is a baby frog, but in this case it is a human. All right, now interestingly enough, if you look at the embryo of different animals, they all look the same at the beginning. Right? And this is one of the things that leads us to believe that we all have a common ancestry, which is that if you look at a human embryo versus a bird embryo versus a reptile embryo versus an amphibian, remember reptiles are snakes, and amphibians are frogs, and then fish. You look at all these embryos, they all look the same during the first few weeks of development, and they only develop to look differently during the latter part of the embryogenesis. Okay, if you look at the human embryo alone here, we measure the age in weeks, which is shown across the top. So during the first 3 weeks, we go from a blastula to an embryo. And during the first 3 or 4 weeks, we have the development of the central nervous system and the heart during the first 3 weeks. We have the development of the eye and the limbs during the 4th week and 5th week, right? And so this stuff is called organogenesis because the organs are forming during the first 4 or 5 weeks. Organogenesis. And during organogenesis, the developing baby, the developing embryo, is particularly vulnerable to having things go wrong. So this is exactly the time during the first 4 or 5, you've heard that it's bad for a pregnant woman to drink alcohol, and that's true. It's bad for a pregnant woman to be damaged, by being hit in the belly with something, mechanical damage. It's bad for a woman to smoke during the first part of pregnancy. It's bad to be exposed to radiation during that period. All of this is true because embryogenesis is developed, the organs, organogenesis, the organs are developing during the first 5 weeks. So this is a particularly vulnerable time for the developing child, is during the first 4 weeks. And oftentimes a woman may not know that she's pregnant for the first 2 weeks. The first 2 months rather. So during the first month, a woman may not even realize she's pregnant. So organogenesis occurs during the first 5 weeks. That's probably the most vulnerable time for the developing baby. So that's why it's a little bit tricky to be a woman, because it is bad to drink or to smoke or to be exposed to radiation during the 5th month of pregnancy, but it's not nearly as bad as it would be during the first 5 weeks, because that's when the most development and differentiation of cells going from general cells to specialized cells, that's when all of that takes place. So that's a particularly vulnerable time. Okay, now we go on to 9 weeks, 16 weeks, 36 weeks, and then full term is 38 weeks or 9 months. Then the baby comes out and we rename it, we change the name from fetus to neonate. What happens at 27 weeks or 7 months? Do you remember when we talked about the respiratory system? This is the period when the type 2 cells in the respiratory system in the lungs, the type 2 cells begin to make surfactant. If a baby is prematurely born prematurely prior to 7 months, they may suffer from infantile respiratory distress syndrome. Okay, now the baby comes out, we have a neonate. So let's just summarize what we learned so far. We learned about the process of fertilization and the process of development after fertilization. So we learned that an ovum is the technical term for the egg, and spermatozoa is the technical term for the sperm. They are both haploid cells. This is what we call gametes, because so humans are normally diploid. They have 2 copies of every gene, 2 copies of every gene in the human genome, and in every cell, in every single cell, except the gametocytes, the gametes. So the word gamete refers to haploid cells that have only 1 copy of each gene and only 1 copy of each chromosome, because that way if you take 1 set of chromosomes from your mother and 1 set of chromosomes from your father and you put them back together again, then you once again create a diploid human being. So the ovum is the fancy word for egg. It is the female gamete. The spermatozoa, a spermatozoa is the gamete for men. It is again a haploid cell. So when a spermatozoa and an ovum gets together, the spermatozoa generally has to swim a long ways. It has a tail. It makes contact with the female egg, with the ovum. And then the head, which carries the chromosomes, which carries the genes, is the only part of the sperm that goes into the egg. And once one single sperm gets into the egg, there's a chemical reaction that takes place that changes the outer surface of the ovum to basically harden it to prevent any other spermatozoa from getting inside, because we only want one spermatozoa, the head of one spermatozoa, to fertilize 1 egg, so that you'll end up with exactly 2 copies of each gene after that. All right, so once you have a fertilized ovum, we rename it as a zygote. So a zygote is a fertilized egg that now has 2 copies of every chromosome. By the way, there are 23 chromosomes in the human genome, so that means you have 46 when you have a fertilized zygote. The zygote then begins to divide, so then you have 2 cells, and then you have 4, and then you have 8, then you have 16, doubling the number of cells every time, until you get a soccer ball, which is called a blastula. The soccer ball then pinches inwards on itself to become a gastrula. That thus begins the process of gastrulation. And then eventually it starts to differentiate or specialize. The cells, basically the cells in a blastula and a gastrula are all the same. So those are what we call embryonic stem cells, even though it's not an embryo, but we call them embryonic stem cells because they're kind of in the early stages, the cells are a generic cell. They're a general type of cell. They do not have any specialty. But as you know, adult humans are made of specialized cells. So the cells of the heart are different from the cells of the brain, and the cells of the brain are different from the cells of the kidneys. And all of these cells are specialized. The process of going from a general cell into a specialized cell is called differentiation. Differentiation. So we start out with a zygote, then a blastula, then a gastrula. These cells are all the same. And then as soon as the cells start to specialize, you see the beginning of the various organs, and you have an embryo. So you have an embryo. And then the embryo continues to develop until you have a fetus, which is basically a small human being. And when the fetus comes out, we rename it as a neonate. All right, let's continue now and look at fertilization, and where is the egg fertilized and how does the egg develop. So where does fertilization take place and where does the egg develop? So I'm going to once again compare human anatomy and physiology to the anatomy and physiology of other animals on Earth, because as I've said before, I think you can learn a lot more about human biology when you compare it to other animals on Earth. You get a fuller appreciation of human biology when you take a brief look at other types of animals. So let's see how the human egg is fertilized versus the eggs of other animals being fertilized. All right, so first of all, is the egg fertilized inside or outside of the animal? A female, of course. Usually we're talking about a female in this case, but as you'll see, we have exceptions. So oviparity, internal versus external fertilization, that's one thing. And then we have, where is the egg fertilized, inside or outside? If the egg is fertilized inside or outside, we can also ask other questions and have other situations. And so these situations include oviparity, which is where the fertilized egg develops outside of the mother. And these eggs, like a chicken egg, which you're all familiar with, chicken egg has a yolk inside, a yolk. And the yolk is actually food that is meant to be absorbed by the developing chicken embryo. So as it turns out, there are a lot of animals that fertilize the eggs either on the inside or the outside. But once the eggs are fertilized, the eggs will be laid in the outer environment and they will develop in the outside. So obviously birds are a good example of animals that do that. Now, ovoviparity is where the eggs are, the fertilized eggs develop inside, but they're sort of disconnected from the mother. The mother retains the eggs, but she's not actually attached to them. So the eggs are inside the mother, but they're still using the yolk sac, or they're using a yolk as food. And then finally, viviparity is where you have fertilization on the inside, and then the egg completely develops on the inside, and as the rudimentary animal is developing, it receives nutrients from the mother. It is not living off of a yolk. So humans, for instance, do that. You don't see a yolk inside pregnant women. They are getting nutrients from the mother's blood supply. Okay, so here's an example of, these are two frogs, which are amphibians, and they're mating, of course, and the male is on the left and the female is on the right. It looks like they're mating the usual way, but in fact it's a little bit different than what humans do. What the male is actually doing here is he's squeezing the female to squeeze the eggs out, and once the eggs are out on the water, the male sprays sperm all over them. So there are a lot of animals, believe it or not, that do it that way. And so those eggs will develop using oviparity. They develop outside the body. Here we have some reptiles. These are turtles. In the case of the turtles, the eggs are fertilized internally. In the case of the frogs, the eggs are fertilized externally. In the case of the turtles, the eggs are fertilized internally, but then the mother lays the eggs, buries them basically, and they develop. So we have oviparity again. All right, here we have some mammals. Rabbits are mammals, and they're mating exactly the way you would guess. We have internal fertilization, where the eggs are fertilized inside the mother, and then they continue to develop inside the mother, and they are dependent upon the mother for nutrients. So the mother's blood supply brings nutrients to the developing rabbits, the developing rabbit pups. And so we have oviparity versus viviparity. Humans, of course, practice viviparity, where similar to the rabbits, we are mammals as well. Rabbits are mammals and humans are mammals, so we do viviparity. So we have oviparity, oviparous development, ovoviparous development, and viviparous development, which are, so you either have internal or external fertilization, and then you have either oviparous, ovoviparous, or viviparous development. All right, now the second question is, how many times does an animal mate during its lifespan normally? So there are many animals in the world, including most conspicuously the arthropods. So the arthropods include the insects, the crustaceans, and the arachnids. Insects, spiders, crabs, lobsters, and so on, where the female who carries all the eggs mates once. That's called semelparity. The female mates once, and then she spends the rest of her life, either she lays a bunch of fertilized eggs at once, or she spends the rest of her life laying the eggs one at a time, and they develop one at a time. Right, so you know, of course, or many, you may or may not know, that bees, for instance, a queen bee is born in a hive. She takes off into the air being chased by a couple of drones, male drones, and once one of these drones mates with the queen, he will fertilize probably 2/3 of the eggs that she's carrying, and then she will go away and she'll form a new bee colony. She'll form a new beehive, and she will continue for the rest of her life laying either fertilized or unfertilized eggs that will develop into adults. And yes, there are some animals where unfertilized eggs actually develop into adults, and bees and wasps are one example of that. That's something that's called parthenogenesis. You'll learn all about parthenogenesis if you take Biology 234. But anyway, so the insects and the crustaceans and these other animals, typically they mate once. All of the eggs are fertilized at once, and then the female either lays them all at once or she lays them one at a time and they develop. But the critical thing is that all the eggs, she mates once and that's it. And all the eggs develop as a result of that single mating. The word idioparity means you mate once and have offspring, and then you mate again if you want to have more offspring. And humans do that. Most mammals do that. So iteroparity comes from the word iteration, which means one time. Right? So or repeating once again. So when you say reiterate, that means do it one more time. You say the second iteration, the third iteration. So the word iteration is the root word for iteroparity. Okay, so here we have a lobster, which is a crustacean. A crustacean is a member of the arthropod phylum that I just told you about. And here's a female that these eggs have all been fertilized at once and they're all developing. So she's ready to, these eggs are all developing at the same speed, which is not something that bees do, for instance. But in this case, her eggs are all fertilized at once, and they're all developing at exactly the same speed, and all of these eggs are going to basically hatch simultaneously and release thousands of tiny, microscopic almost, very, very tiny lobsters into the ocean. If you take Biology 110, you'll learn that's one strategy for ensuring that your species survives is to produce a lot of them, a lot of offspring. But very few of them survive because when they start out, they're small and their shell hasn't developed, and they tend to, most of them, 90% of them probably get eaten by fish. So most of them having large numbers of offspring that are helpless at the beginning is one way of ensuring survival, that, you know, predators can't eat them all. So anyway, that's a characteristic of animals that do semelparity versus mammals, which do iteroparity. So the other types of animals, like mammals, for instance, do iteroparity. So humans, every time we want to make children, we have to mate again. Separate matings every time that you want to have children. Okay, some of the mammals produce a lot of offspring. You know, humans, there are some mammals that produce only a few. Typically the higher predators produce only a few, one or two at the most per mating. But there are some animals like dogs and rabbits that produce more than quite a few, like a dozen or so. Okay, so we have dog pups in the previous picture, and do you know what the word is for baby rats? It's pups as well. So baby rat pups are shown here. All right, now humans, as I said, humans appear to be designed or adopted to having only one child per cycle, per what's known as a menstrual cycle. But occasionally you get a woman who gives birth to 2 children, or 3 children. 2 children we call twins, 3 children we call triplets, 4 children we call quadruplets. But a few years ago there was a famous woman who gave birth to 8 babies at once, and she already had 2 children, so she had 10 at that point. So her actual name was Nadia Suleiman, and she was dubbed by the media, she was dubbed the Octomom, octa meaning 8. And this did not happen naturally. It happened because of some mischief was going on with artificial insemination. She bribed a doctor, a fertility doctor is somebody who helps couples who can't have children to have children. They sometimes do something called in vitro fertilization. In vitro is the Latin word for in glass, and so in glass means in a test tube. So if you mix eggs and sperms together in a test tube and wait until they start developing, and then you put them into a woman so that she can carry the baby to term, that's called in vitro fertilization, because the fertilization did not happen in the mother. If it happened in the mother, that would have been called in vivo fertilization, but it happened in vitro in a test tube. And so normally a fertility doctor is only supposed to implant 5 or so of those fertilized eggs back into the mother, in the knowledge that only of the 5 that you put in, only 1 or 2 will actually stick, as it were. Only 1 or 2 of them will actually develop. This woman apparently bribed or convinced the fertility doctor to put in a lot more than they were supposed to put in. So instead of giving birth to 1 or even 2 babies, she gave birth to 8. And I'm not exactly sure why she would want to do that, because it's quite a danger to your health. And here you can see why. If you're carrying 8 babies, that is a very dangerous situation for your health. So generally, it's not healthy for a woman to carry more than 1 child at a time. 2 is a bit of a health risk. 3 is a bit of a health risk. So you always have to be careful. Certainly 8 is very ill-advised. Okay, so this was done by in vitro fertilization. Okay, to summarize, there's a difference between heteroparity and simulparity. There's a difference between internal fertilization and external fertilization, and then the development, where the development takes place, is what gives us the words oviparous, ovoviparous, and viviparous. And of course, this is what humans do. Humans practice heteroparity fertilization, they have internal fertilization, and they have viviparous development. All right, now you know, of course, that humans and all mammals, in fact, have two permanent sexes. That is, they have a male and a female. Generally, the male just produces the sperm, and then the female produces the eggs, as well as carrying the developing child. Not all animals do that, of course. But nevertheless, in the case of mammals, it's the female that carries the baby. So there's a male and a female, permanent sexes. Believe it or not, there are many animals in the world that can be born one sex and then change sexes depending on what's going on in the environment. So for instance, frogs and fish can have the ability to be born, and some other animals have the ability to be born as a male or a female, and then if it's perceived that you need more females, there are too many males, you need more females, some of the males can turn into females, or the other way around. If there are too many males or too many females, some of the females can change into males during development as a result of hormones. Humans don't do that. Naturally, anyway. Humans don't do that naturally. There is artificial means called gender reassignment surgery, where you change the gender of somebody surgically, and then you follow that up with hormones from the opposite sex until they turn into what is essentially a fairly good copy of the opposite sex, but never really exactly the opposite sex. But anyway, so what is it that determines whether you're male or female under normal circumstances? And do we really need male and female? Well, as I said, some animals don't have that, but mammals do. Most of the more developed animals do have 2 sexes. All right, so do we really need 2 sexes? Well, there are some organisms that are asexual, and they reproduce through what's called binary fission. That means they simply split in half. The more primitive organisms do that. Bacteria do that. Many protists do that, protozoa. They simply split in half. As I mentioned at the beginning of the lecture, that does not lead to much genetic diversity. If you don't have genetic diversity, then evolution proceeds very slowly. If you have sexual reproduction, evolution proceeds very rapidly, and then you have evolution happening quickly. Okay, so there are some organisms that are basically asexual and they reproduce asexually, but there are other organisms that are hermaphrodites. A hermaphrodite is an organism that has both male and female sex organs. So a hermaphrodite has both male and female sex organs. Most plants, or many plants, are hermaphrodites, and members of the annelid phylum, the phylum called Annelida, which is a type of an organism that has its own group, its own phylum, it's called. And these are the worms, basically. The phylum Annelida, these organisms are commonly referred to as worms. So worms basically have two sexes. They're hermaphrodites. They have male organs at one end of the worm and female organs at the other end of the worm. And so they can mate with another worm and exchange genetic information. But if they can't find another worm, they're perfectly able to mate with just themselves and produce lots of offspring, but with less genetic diversity. So here we have a plant, a typical plant. These are angiosperm plants. So we have male gametes being produced in the stamens, and then in the ovaries we have eggs that are produced. The pollen fertilizes the eggs. Basically, it sends a fertilization tube down the pistil and fertilizes the eggs. So a plant is capable of self-fertilization, but it's more efficient for it to fertilization to take place in between plants, and that of course is carried out by third parties like bees, for instance, carrying pollen from one flower to another and thus spreading the genetic diversity around the plant kingdom. And as I mentioned, hermaphrodites, the annelids, the worms, are frequently hermaphrodites, so they have male sex organs at one end and female sex organs at the other, so they can mate with another worm, with another annelid of the same species, or they can simply turn around form a U shape and mate with themselves. All right, now if you do have different sexes, the information which determines whether you're one sex or another is probably contained on what are known as the sex chromosomes. So I mentioned earlier on in the course that chromosomes are, an individual chromosome is a long piece of DNA that generally contains 1,000 or so genes. Humans have 23 chromosomes, approximately 1,000 genes per chromosome, and we number the chromosomes 1, 2, 3, 4, 5, 6, 7, 8, 9, and so on from up to 22. And the chromosomes that don't determine which sex we are referred to as autosomes. Autosomes carry all of the genes that have nothing to do with what sex we are. There is usually a pair of chromosomes that are called the sex chromosomes. In humans, that is chromosome set number 23. And you know, of course, most of you know that males have the chromosomes are designated as being either X's or Y's, because they kind of look like an X and a Y under a microscope. So if you have two X's, you're female. If you have an X and a Y, you're male. And so it's actually the presence of the Y chromosome that determines if you're going to be male or not. If you don't have the Y chromosome, you end up developing into a female by default. And you develop into a female by default. Okay, so X is actually a regular chromosome, and the Y chromosome is not really a regular chromosome. It's a little chromosome that has only a few genes on it, and those few genes are involved in determining whether you're a male or a female. If you have the Y chromosome, you will turn into a male. If you don't, you'll be a female. Okay, so as you know, XX, if you have XX chromosomes, you're female. If you have XY, you're male. Okay, now the Y chromosome, the Y chromosome is not really a chromosome. It's an abbreviated chromosome that has only a few genes involved in sex determination on it. So if there's a mishap, there's a misfire during what's known as gametogenesis. Gametogenesis means the formation of gametes, or the formation of haploid cells that are used in reproduction. So in other words, when the female eggs are developing in the ovaries, or when the male sperms are developing in the testes, occasionally there's something goes wrong, and so the sperms and the eggs that are produced may not get all of the chromosomes that they're supposed to have. They're supposed to have a complete set, but occasionally you have a misfire, you have an accident, and you have a sperm or an egg that's produced that's missing one of the chromosomes. Or occasionally you have a sperm and an egg that has one too many. Right, so you're supposed to have one copy of each chromosome. If there's a mistake, you actually have two in an egg or in a sperm. That means that if that sperm that has 3 of chromosome number 22, for instance, if you have chromosome number 22 in a sperm, and that sperm ends up connecting with an egg that has one of its own already, that means that will give rise to a human being that has 3 copies of chromosome 22 instead of 2 copies. They'll have 2 copies of every other chromosome and 3 of chromosome 22. And in genetics, we call that trisomy. So trisomy. But occasionally you have a misfire or an accident where you end up with a sperm that has you have an egg that has no X chromosome, and then you have a sperm that has a Y chromosome. So they come together and you have Y and nothing, right? The O here, YO, the O indicates nothing. It's a genetic, an abbreviation that geneticists use to indicate that there's no other chromosome that matches with the Y. So as I said, the X chromosome is a regular chromosome, the Y chromosome is not. And so if all you have is the Y and you don't have an X, you are non-viable. Non-viable means you cannot live. Non-viable. So viable means you can live. Viable comes from the word via, which means life. And so non-viable means if you have a Y chromosome and no X, you cannot survive. Okay, what about an X? I said that the X is a regular chromosome. What if you have an X due to an accident during the creation of gametes? What if you have an X and no other X? Or what if you have an X and no Y? That would be XO. Right? So what would that be? Well, as I said, if you don't have a Y, then by default you will become a female. Right? So there are women in the world who have an X chromosome and no other X chromosome or no Y chromosome. And they're female. They have developed into females. Right? So they have 2 copies of every other chromosome, but they only have one X and nothing to match it with, but they develop into females. They develop into women. Okay, so interestingly, insects don't do that. So in the insect world, if you have a Y chromosome, you have a Y chromosome, you're a male, but if you're missing the Y chromosome, you do not develop into a female by default, you develop into a male by default. So I mentioned earlier that parthenogenesis is something that bees and wasps and moths do. It means that, let's take the example of a bee. If a queen mates with a drone, the drone will fertilize not all of the eggs, but probably 2/3 of them or something like that. So the 2/3 of the eggs that were fertilized become females, and they are diploid. And the 1/3 or the 1/4 that did not get fertilized, the queen will still lay those eggs, and those eggs will still develop, but they will not be diploid, and they'll develop into males. So male bees are called drones, and so drone bees are actually haploid. They only have 1 copy of every chromosome, which is kind of unusual. It's in the animal world, it's quite unusual for a haploid organism to develop. They're usually at least diploid. Some animals have more than 2 copies of each chromosome, but at least they have to have 2 copies. Okay, so in mammals, which is what humans are, of course, in mammals, the presence of the Y makes you male. So XX is female, XY is male, X nothing is female, and that leads to a condition called Turner syndrome. So this is something I might ask you on the final exam. What is Turner syndrome? So Turner syndrome is where you have X nothing, XO, and that person develops that when you have, I mentioned earlier that you're supposed to have 2 copies of each chromosome. If you accidentally have 3 copies of one of the chromosomes, that's referred to as trisomy. You might have heard of Down syndrome, which is called trisomy 21 because it's chromosome number 21 that you have 3 copies of. And on the other hand, if you have only 1 copy of a chromosome instead of 2, that's called, what would you guess? What would you guess? It's actually called monosomy, right? So monosomy. So there is a condition called Turner syndrome where you have monosomy of the X and you turn into a female. There are cases where you are trisomic for the X. You have 3 X's. You're trisomic for the X, and you are physically female as well. That's referred to as triple X syndrome. And there is a condition where you have 2 X's and a Y, and I said that having the Y makes you male, and that is referred to as Klinefelter Syndrome. Right? So here we have the 2 normal conditions, male and female, determined by XX and XY. That's what most people have, but there are exceptions. X nothing, Turner syndrome. 3 X's, triple X syndrome. And XXY, you're male and you have Klinefelter syndrome. But this again illustrates that if you have no Y, you become female by default. Okay, so this by the way is these are the symptoms of Turner syndrome. This is so Turner syndrome, X nothing, XO. You are in appearance you're female, right? Women with Turner syndrome, they have a round heart-shaped face. That's one of the characteristics. They have a large, what's known as a shield chest, a big chest that's shaped like a shield that a knight used to carry in the old days. There might be some problems with the aorta. Coarctation of the aorta is coarctation of the aorta just means there's a little, the aorta is a bit pinched, so it's difficult for blood to circulate, so they may have some minor heart problems. Women with Turner syndrome tend to have a shriveled uterus and shriveled ovaries, so they're sterile, can't have children. And probably and they tend to be short, on the short side, 5 feet or less. And one of the most obvious characteristics is the presence of a webbed neck. The neck, instead of going straight down, it kind of goes like this. Right? You can see the webbing of the neck. Now, typically, if a girl is born with Turner syndrome, her parents may elect to try and, sadly, we live in a very cruel society where people make fun of children, pick on each other if they're different, right? So you have children that pick on or bully children that look different. Sadly, we haven't, that's bad. We try to discourage that habit, but it does still happen. And so for that reason, some parents that have Klinefelter syndrome daughters decide to try and mask the effects of the Klinefelter syndrome. So there are 2 ways that you can do that. One way is you give the girl large doses of female hormones during adolescence so that she develops a more of a typical female shape, wider hips, narrower waist, and better breast development. And they do cosmetic surgery, plastic surgery, to remove the webbing from the neck so that they have a normal looking neck. Right? So those are some of the options that are available to people who have Turner syndrome. All right, triple X syndrome has no symptoms, no physical symptoms that we can tell, except the women tend to grow a little taller if they have triple X syndrome. And so they tend to grow into have a tall, lanky appearance. And so there's probably slightly a higher percentage of runway models, that are, typical high fashion models that have triple X syndrome, and female basketball players sometimes that grow very tall. But other than that, there's no way to tell. So there are because there are no obvious symptoms, there are a lot of women walking around in the world who probably have 3 X's and don't know it. It's just called triple X syndrome, but it has no unusual effects on the physique or the phenotype. And as I said, finally, Klinefelter syndrome is where you have a male that has two X's and a Y. So instead of XY, he has XXY. He has male genitals, but they tend to be shrunken. He has what's known as gynecomastia, which is breast development, minor breast development in the male. Tends to have lack body hair, no body hair, and a high forehead. Other than that, other than those slight physical differences from a normal male, there's no, there are no other symptoms, no other real symptoms to Klinefelter syndrome. Okay, so to determine about human and mammal sex determination, we have autosomes. Those are the chromosomes that carry all the genes that are not involved in sex determination. And we have one set of sex chromosomes, and then we have XX versus XY, as well as some anomalies. Right. Okay, let's move on now from looking at what happens in the animal kingdom and so on, and let's specifically talk about the mechanics of the male and the female reproductive system, the male and the female reproductive anatomy. Okay, so the male sperm is produced in the testes, and if you open up testes, what you would find is a big ball of thread. It looks like a ball of thread inside the scrotum, as it's called. So if you look at a testy, you have this ball of thread, and these threads are called seminiferous tubules. Seminiferous tubules. And the sperm are developing inside the seminiferous tubules. They are sent from the testes to the penis through a tube called the vas deferens. And we're going to talk about male reproduction and female reproductive functions. And so and also birth control and contraception. We'll get into that topic in this lecture. But because the semen or the sperm is sent to the penis through a tube called the vas deferens, there is an operation that men can have to make them sterile. If a man deliberately does not wish to have children, does not wish to father children, he can have an operation called a vasectomy. So a vasectomy is where a surgeon cuts the vas deferens and ties it in a knot so that the sperm can't get from the testes to the penis, right? So then that man can no longer father children. In fact, what happens is the spermatozoa are simply absorbed in the walls of the vas deferens. It's not like they back up and the vas deferens breaks or bursts or something because the sperm cells have backed up, or because there's a blockage there. They actually get absorbed. And so but nevertheless, they can't make it to the penis, and therefore they can't be used to fertilize eggs. So one of the operations, an operation that a man can have, is called a vasectomy, which is where the vas deferens, there are two of them, one on each side. There's one vas deferens that goes from the testy to the penis on each side. So there are two testicles, and then there are therefore two vas deferens. Okay, now the vas deferens, on the way to the penis, they have to pass through a large gland called the prostate gland, and some other glands that mix various lubricants with the spermatozoa. So when the spermatozoa are produced in the seminiferous tubules, they are called spermatozoa. When they get mixed with these lubricants inside the prostate gland and the other glands, they are collectively, the spermatozoa and the lubricants are referred to as semen. And then the semen is sent out through the urethra, which exits out of the penis. Now you may have heard the word urethra before. So in the male reproductive system, both urine and semen are sent out of the penis through the urethra. So in females have two different openings. There's a vagina for the sexual function, and then there's a separate urethra that comes out through a structure called the urinary meatus. So in the male system, both the urine and the spermatozoa come out of the same tube, the urethra. In women, there are two separate tubes that fulfill either the urination function or the sexual function. All right, now we're going to talk about like we do with every other organ system. We'll talk about pathologies or malfunctions of the different systems. So there are some malfunctions. There are two different pathologies of the male reproductive system that I'd like you to know about. One of them is a very common condition in older men, which is called erectile dysfunction. And what's going on there is that within the penis, obviously the penis has to become erect in order to fit into the vagina and fertilize the eggs, right? So the shaft of the penis is filled with a number of blood capillaries, and the bloodstream, we have, like all capillaries, there's something called a pre-capillary sphincter, which opens and closes to determine whether blood actually goes into that capillary or not. As men get older, those pre-capillary sphincters begin to malfunction, which makes it difficult for them to get an erection, and that's called erectile dysfunction. But the penis has erectile tissue, which is basically capillaries that fill up with blood in order to erect it. If the pre-capillary sphincters malfunction, then older men often get a condition called erectile dysfunction. So in fact, probably in men over 50, it's probably about 40%. The rate of erectile dysfunction in men in their late 40s, early 50s is around 40%. So it is a fairly common problem. It's not always a complete problem. It's just something that recurs every now and then. And so if you watch television programs that are targeted to a male audience, like sporting events or news or something like that, you'll see that those programs, if their target audience is older males, older men, they will just flood those programs with television commercials advertising Viagra and Cialis. And Viagra and Cialis are two popular drugs that are used to treat erectile dysfunction. So they basically allow the erectile tissue to fill up again. So that's a target audience for television commercials. These television commercials are just on all the shows that are targeted to, all the television programs that are targeted to older men. Okay, another problem with the male reproductive system is something called an enlarged prostate. And so the prostate gland, which is involved in the reproductive system, the urethra passes through it. And remember that both semen and urine come out through the urethra at different times, obviously, but they both come out through the urethra. Now, if the prostate gland swells up, it pinches the urethra shut, which makes it difficult for the man to urinate. And so again, probably about 40 or 50% of men over 50 have a problem with an enlarged prostate, which means that they can't urinate properly. If they try to urinate, the bladder doesn't empty completely, and so then they end up having to urinate twice as often, but only half as much comes out because the bladder is not able to completely empty because the prostate gland is kind of pinching the urethra shut. Okay, so those are two problems, two pathologies of the male reproductive system: enlarged prostate, erectile dysfunction. All right, here we have a diagram. So here we have the testes down here, and we have the seminiferous tubules in each of the testes. Right? And then there is a vas deferens, this tube that carries the spermatozoa around behind the bladder and then through the prostate gland, which is located just underneath the bladder, to the urethra, and then out through the penis. This is the shaft, which is filled with erectile tissue, which are basically blood capillaries. This part in the front is referred to as the glands. And so that's the male reproductive anatomy seen from the front. Looking at the male reproductive anatomy from the side, we have the testes, which are filled with the seminiferous tubules. Then we have the vas deferens, which again goes around over and behind the bladder, the urinary bladder, and then through the prostate gland, out through the erectile tissue in the shaft, and out through the glands. Okay, here we have a close-up of the seminiferous tubules. So here's a testis, and if you pull out one of these threads and you cut through it, you do a transverse section so we can see what's inside. You would see this. It's about, as I said, it's about the size of a thread. And what's happening is that we have diploid cells around the periphery of the seminiferous tubule, and they move towards the lumen in the center. They move towards the hollow space in the center, and as they move towards the lumen in the center, they divide in half, become haploid cells through a process that's called meiosis. Regular cell division is called mitosis, and then production of gametes is called meiosis, where the cells divide twice without copying the genome. We won't really go into that in this course. But as they move towards the lumen, towards the center of the seminiferous tubules, they develop a tail and they become haploid. And then these sperms can swim because they have a tail. They swim up through the vas deferens and out through the penis. All right, so here we have on the outer surface of the seminiferous tubule, we have something called a spermatogonium. You should probably remember that word. And the spermatogonium undergoes 2 rounds of cell division called meiosis 1 and meiosis 2. And during those rounds of cell division, the genome is not copied. So this thing here that says 1N versus 2N, 2N means that you have 2 copies of the genome and you're diploid. You're a cell that has 2 copies of the genome. And 1N means that you're a cell that has only 1 copy of the genome. Right, so the N number refers to the number of copies of the genome that you have, which is also the same thing as saying the number of each chromosome pair that you have. So if you have only 1 of each chromosome, you're a 1N cell. If you have 2 of each chromosome, you're a 2N cell. Nevertheless, when you get to the center, you have a tail, and you're a fully functional spermatozoa. Now let's take a close look at the fine structure of a spermatozoa. So we have a tail, of course, right? The tail whips around and swims. Remember, we said that the energy inside a cell is created by the mitochondria burning glucose and converting it into ATP. So this tail is powered by ATP, just like any other movement in the body is. And so in the mid-piece, so we have the tail, and then we have the mid-piece. Within the mid-piece, we have about a dozen mitochondria who are there. They're like the jet engine that's powering that tail. And so those mitochondria that have been packed in there are there to power the tail. Then in the head of the sperm, we have the nucleus from the original male cell. So that nucleus contains 1 copy of each chromosome, not 2 copies, but 1 copy of each chromosome. It is therefore a haploid nucleus. And at the very front of the sperm, we have something called an acrosome. Acrosomy means to attack or to assault, right? So this is a hostile, aggressive word, acrosome. And what it actually is it's a bag filled with enzymes that allows the sperm head to penetrate the egg, right? So it's like the explosives in the end of a torpedo or something. It allows the swimming sperm to hit the sperm will hit up against the egg, and then the acrosome will break open and allow the enzymes out, which allows the head to go into the egg. But only the head goes into the egg, the mid piece does not, which means that now you've got the head from the sperm inside the egg. We reclassify it as a zygote. It's no longer an ovum, it's now a zygote. And only the only mitochondria that you find inside that zygote came from the mother. The father's sperm left his mitochondria outside. So that's why I mentioned earlier that you might have heard of something called mitochondrial DNA. This is when you're trying to do a genetic fingerprint to determine who somebody's parent is. One of the ways that you can determine unambiguously, you can determine for certain who somebody's mother is by looking at the DNA in their mitochondria, because the mitochondria inside the fertilized zygote came from the mother. The father's mitochondria were left outside. So that means that each of us has mitochondrial DNA that's identical to our mother, who has mitochondrial DNA that's identical to her mother, that's identical to her mother, that's identical to her mother, and so on. So one of the ways that you can determine unambiguously, that you can determine for certain who somebody's mother is and somebody's maternal grandmother is and who somebody's maternal great-grandmother is, all you have to do is look at do a genetic fingerprint on the mitochondria. All right, so here we have some spermatozoa attacking an ovum, but the first one to shove its head inside the ovum will successfully start a chain reaction, a chemical chain reaction that prevents all the others from getting inside. So that's a very successful way of making sure that each human that's created is diploid, because the egg cannot be fertilized by more than one sperm head. Okay, so to summarize the male reproductive system, the spermatozoa are produced in the testes and sent to the penis through they're produced in the seminiferous tubules and sent to the penis through the vas deferens. A vasectomy is an operation that men get to become sterile because they don't want to have children. The vas deferens has to pass through the prostate gland. If the prostate gland is swollen, that makes it difficult for men to urinate. So a very common problem that older men have is called an enlarged prostate. You will see commercials, again, any television programs that are targeted towards middle-aged men or older men. You will also see all of these advertisements for vitamin supplements called super beta prostate, for instance, and those are vitamins and minerals that are supposed to be good for relieving the swelling of the prostate and making the good prostate health, basically, to try and reduce the amount of enlarged prostate problems. Okay, then the penis itself has capillaries that provide the erectile tissue. If they malfunction, then the man has something called erectile dysfunction. And Viagra and Cialis are two drugs that are meant to counteract that. Okay, so as I mentioned, spermatogenesis, the word genesis means the origin of or the beginning of, so spermatogenesis means the beginning of what is the source of the spermatozoa. So what you do is you start out with one diploid cell called a spermatogonium, which is located on the periphery of the seminiferous tubule, and when it wants to replace itself to make sure that you can keep on making sperms, it divides the normal way, which is called mitosis, rather. So mitosis refers to regular cell division. Meiosis refers to two successive rounds of cell division where the genome is not copied in between, so you go from having two copies to one copy during the second phase of meiosis. So meiosis 1, you end up with two haploid cells called spermatocytes, and then during meiosis 2, you end up with 4 spermatids, what are called spermatids, and then so you end up with, basically, you end up with 4 spermatozoa for every spermatogonium that you started with. The production of spermatozoa in males begins at adolescence and continues until death. You'll see for women, of course, women don't produce eggs until old age. Women go through a phase called menopause, which is the period where their brain stops making hormones that tell their ovaries to produce or to allow the eggs to mature. So men continue producing spermatozoa through beginning in adolescence and continuing throughout their whole life, whereas women begin to produce eggs in adolescence and generally stop somewhere around age 50, approximately age 50. Now let's move on to the female reproductive anatomy. So the female version of the gonads, the word gonads is a generic term meaning either the testes or the ovaries. The testes in men or the ovaries in women. They are collectively referred to as the gonads, and it's the same for other animals as well. The gonads refers to the organs that produce the haploid sexual reproductive cells. So in a woman, you start out with a diploid cell called an ogonium, and it undergoes 2 rounds of meiosis. And what it does is, unlike the male spermatogonium that ends, in males, if you have 1 spermatogonium, you end up with 4 spermatozoa. In women, if you have 1 ogonium, you actually end up with only 1 functional ovum and 4 other things that are called polar bodies that are duds, basically, that didn't work. Okay, so now this is critical. The oocyte is a premature ovum, right? So a developing ovum is called an oocyte. The oocyte is stored in a structure inside the ovaries called a follicle. Right? So you've heard the term hair follicle. A hair follicle, of course, is a cluster of cells that work together to produce a hair. Right? Now, a follicle in the ovary is a cluster of cells that work together to cause the development of a mature ovum. So what the follicles are, you have a developing oocyte in the center, and surrounding that developing oocyte, you have a bunch of other cells that are referred to as nurse cells, N-U-R-S-C, nurse as a hospital nurse, or nursing a baby. A nurse is somebody who takes care of a baby. So we call those nurse cells, and those nurse cells form a follicle, which is basically where the oocyte continues to develop. Okay, now so the female gonads are the ovaries. In the ovaries, a woman is born with thousands of follicles, right? And then those follicles will begin to develop into mature ova in adolescence, and they will continue to develop until menopause at approximately age 50. Now, they do not all develop at exactly the same rate. So when a woman is born, you've got thousands of these oocytes in follicles inside the ovaries, and they will continue to develop at different speeds so that one becomes mature and is released during a menstrual cycle, and then another one will become mature. Hopefully we don't get more than one maturing at exactly the same time, because then you'll give birth to twins, and that can be dangerous. Hopefully you don't have three that are maturing at the same time. Generally they don't. Generally, they're all developing at a slightly different stage, right? So one of them becomes mature before the others, and it gets released, and that forms a menstrual cycle, and then another one becomes mature and it gets released. It's rare that two will mature at exactly the same time, or three will mature at exactly the same time. So here we have an ogonium, and we have the two rounds of meiosis that give rise to one mature ovum. It gets fertilized by a sperm, and then we have a diploid zygote. Okay, so what happens is that the ova, ovum is singular, ova is plural, are produced in the ovaries, the female gonads. They travel down, so women have two ovaries, one on each side, and the uterus, women have two ovaries and one uterus. The uterus, of course, is what becomes the womb when a woman is pregnant, the womb, W-O-M-B. And each fallopian tube is connected to each ovary is connected to the uterus by a fallopian tube, which is also known as an oviduct. Right? So a fallopian tube is a tube that connects the ovary to the uterus, and women have two of them. So the fallopian tube is also referred to as an oviduct, sometimes, oviduct. So fertilization, now interestingly, when men and women have sex, the fertilization does not take place in the uterus. The fertilization generally takes place in the fallopian tube. So that means that the sperm has to swim all the way from the vagina through the uterus down the fallopian tube until it almost reaches the ovary, at which point it will encounter a loose ovum rather. So the fertilization generally does not take place in the uterus, which is where you would expect it to take place. The fertilization actually takes place in the fallopian tube somewhere. So then this fertilized egg develops into a blastula, and it continues to divide as it travels down the fallopian tube. Eventually it reaches the uterus, and the blastula will implant itself into the swollen wall of the uterus. The wall of the uterus, as we learned in an earlier lecture, is called the endometrium. So this fertilized blastula that's continuing to develop will get stuck in the swollen endometrium, in the endometrial wall, and develop into an embryo, and then a fetus, and then a neonate when it comes out. Okay, now between the vagina and the uterus, we have a ring of muscle, a sphincter called the cervix. So its purpose basically is to guard the entry to the, prevent anything bad from getting into the uterus. And then obviously the cervix has to dilate. Dilate, of course, means to open up. The cervix has to dilate in order to let the baby out. So when a woman is giving birth, they talk about dilation. They're talking about the cervix opening up. It kind of opens up in phases, and you can measure how big those phases are until it's big enough to let the baby's head out. The baby's head, of course, is the biggest part of its body. So getting the head out is the hardest part by far. Okay, so then below or inferior to the cervix, we have the vagina. The vagina is basically where the penis goes in order to send the spermatozoa into the uterus and down the fallopian tubes. And then the term for the external folds, like their external folded tissue on the outside of the vagina, is collectively referred to as the vulva. And the clitoris is a central bump of flesh that's located on the anterior part of the vagina. And the clitoris is considered to be part of the vulva. Okay, so the urethra, there is also a urinary, in the same location, in very close proximity to the vagina, we have the urethra, which is where the urine comes out. The urine comes out of a kind of a fleshy lump called the urinary meatus. All right, so here we have a close-up of the female reproductive anatomy viewed from the front. So we have two ovaries, and we have two oviducts, or fallopian tubes. And we have one uterus where the fertilized egg comes down the fallopian tube and implants itself in the endometrial wall. At the bottom of the uterus, we have the cervix, and then we have the vagina, and then outside we have the vulva, the vulval folds. Okay, so notice that inside the ovary, we have some follicles, right? So this arrow is pointing to follicles, these little places where you have nurse cells that are feeding and allowing for the development of ova, and eventually one of them will mature first and will burst its way out of its follicle and start traveling down the oviduct on the way to the uterus. And that follicle, what's left of it, will swell up and turn yellow and become something called a corpus luteum. Learn that word corpus luteum. Learn the word corpus luteum because it's very important, and we'll be discussing it later. Here we have the female reproductive system viewed from the side. Okay, so here's the uterus. Here are the fallopian tubes of the oviducts, and here's the ovary over here. Notice, so the ovary is just slightly superior to the uterus. So we have the endometrium there, we have the cervix, we have the vagina, and then down here on the outside we have the vulva and the clitoris and the urinary meatus, which is right here. Now notice, here is the urinary bladder. Right, here's the urinary bladder, and here's the uterus. So when a woman is carrying a baby, when that developing fetus gets to be very big, there's not much room for the bladder to fill up because the uterus has suddenly expanded in size to carry the baby, and so the woman's bladder doesn't have much room left anymore to fill up with urine, which means that it's not atypical, it's not unusual for a pregnant woman to have to urinate quite often because the bladder doesn't hold very much. There's not enough room for it to hold. Okay, so let's take a closer look at the ovary. Right, so we have follicles inside the ovary, and we have ova maturing inside those follicles. The unfertilized egg is released. It is met in some cases by spermatozoa that were swimming down towards the ovaries, so that an egg is generally fertilized right there. The developing egg continues to develop into a blastula, and then it reaches the uterus and then implants itself into the endometrial wall and starts to develop. So fertilization generally takes place in the oviduct, not in the uterus. Okay, now, when the blastula embeds itself in the endometrium, in the endometrial wall, it suddenly forms a structure called a trophoblast. The trophoblast will become the placenta. So, as you know, a developing baby is surrounded by a structure, by a membrane called a placenta. The placenta is actually created by the baby, not by the mother. I'll tell you a story. My first job, when I first got my first Bachelor of Science degree, my first job was in St. Paul's Hospital. I was a research technician in St. Paul's Hospital, which is kind of near Columbia College. And the lab instructor, or not the lab instructor, but the lab supervisor, was trying to think of an introductory project for me to work on, and she came in one day and put this frozen block of tissue down in front of me and said, "This is a placenta. I want you to find out whether it's male or female." And I thought, I knew that a placenta was wrapped around the baby like a baby develops in a placenta. And I thought, well, listen, what she wanted me to do was she wanted me to extract the DNA from this placenta and then determine whether the placenta had XX or XY chromosomes. And I thought, well, don't they? I said, don't all the placentas have XX chromosomes because they come from the woman? And she kind of said, "Oh, you're an idiot. Don't you know that the placenta is created by the baby, not by the mother?" So the placenta is actually a structure that's created by the baby, and so it starts out as a smaller structure called a trophoblast. Trophoblast becomes the placenta. So as the baby develops, we have this placenta structure inside the uterus. And what happens in the placenta is that the mother's blood vessels and the baby's blood vessels come very close together, so close together that oxygen and carbon dioxide and amino acids and other nutrients and ammonia wastes, nitrogen wastes can be exchanged between the baby's blood and the mother's blood. Most importantly, the oxygen and the nutrients, basically. They can be exchanged, but the blood does not actually contact each other. The baby's blood and the mother's blood does not actually come into contact. And we mentioned earlier that one of the reasons why men and women having babies, if they have incompatible blood types, could be a problem, but it's not as much of a problem as you might think, because the placenta actually prevents the mother's blood and the baby's blood from coming into contact. So if you have a type O mother and you have a type A or a type B baby, it shouldn't be a problem in the first birth, because the mother's blood and the baby's blood don't actually come into contact. Therefore, the white blood cells that are part of the mother's immune system will not come into contact with the baby's erythrocytes, the baby's red blood cells, and the mother's immune system will not destroy the baby's blood. And that's a function of the placenta. Okay, we talked about two problems with the male reproductive system. Let's talk now about two problems with the female reproductive system. Endometriosis is where the tissue that forms the fallopian tubes, endometriosis is where the endometrium grows out into the fallopian tubes where it should not be. So the fallopian tubes are ciliated epithelial cells. The endometrium the uterus is lined with another type of columnar epithelium called endometrium. Endometrium is where the fertilized egg is supposed to implant itself, but occasionally, if the endometrium grows out into the fallopian tubes, the egg could implant there. In any case, even if the egg doesn't implant itself there, remember that during the monthly menstrual cycle, towards the end of a woman's menstrual cycle, the endometrial tissue, the uterine wall, actually swells up and becomes very thick. Now, if this tissue that's lining the fallopian tubes swells up and becomes thick, that makes it more difficult for the egg to actually get through, and that causes painful menstruation. So when a woman who has endometriosis, when she has her monthly period, when she has the monthly bleeding, it's actually quite painful. So that's called endometriosis. Now, we said that a fertilized egg is meant to implant itself into the endometrium. The endometrium is supposed to be in the uterus. What happens if the endometrium grows out into the oviducts into the fallopian tubes. Isn't it possible that the fertilized egg could implant itself in the fallopian tube? And the answer is yes, and when that happens, that's called an ectopic pregnancy. In biology, the word ectopic means in the wrong place or at the wrong time. Ectopic means it's someplace where it shouldn't be. So an ectopic pregnancy is where a fertilized egg implants into the fallopian tube instead of into the uterus, and obviously the fallopian tube can't expand to hold a whole developing baby, and so it usually just breaks it instead. And so a woman will have a broken fallopian tube. So generally if that happens, generally the fallopian tube is damaged and you can't receive any more eggs from that uterus, from that ovary. But at least you've got another ovary and you can receive eggs from that one. If the same thing happens on the other side, you could end up being sterile. In fact, that's what happened to my sister-in-law. My brother's wife had 2 ectopic pregnancies and broke both fallopian tubes, and that's why they were never able to have any children. So that's one of the reproductive problems with the female reproductive system. All right, now let's move into a very complicated subject, which is how ovulation. The word ovulation refers to the production of 1 egg per month. How is ovulation controlled in humans by hormones that are released by the brain? So you know that a woman has a monthly period, and what's happening during that monthly period is that one of the mature eggs has burst out of its follicle. And if it's fertilized, it will implant itself into the uterine wall, into the endometrium, fine. If it's not fertilized, it will just go out and it will flush out all of this blood and tissue that was actually part of the endometrial wall. So every month the endometrial wall swells up and develops new arteries and veins, arterioles actually, which are small arteries, and small veins, venules they're called. Develops these blood vessels and swells up in the expectation that it will be providing blood to a developing baby. So if the fertilized egg doesn't implant, the body doesn't need all this stuff, so the tissue from the endometrium will just come out along with a lot of blood and some other things, right? So that's called the monthly period, the menses. And so this process of happening every month is controlled by hormones that are released from the brain. And let's see how that works. The reason why we do that is because we want to avoid, the reason why we control this process through hormones delivered from the brain is because we want to avoid having octomoms. We want to avoid having more than 1 egg available to be fertilized every month. So the release of 1 egg per month is carefully controlled by hormones from the brain. It's actually a very complicated process, but let's see if we can understand it. Okay. The brain releases a hormone called gonadotrophin releasing hormone, which is abbreviated GnRH, gonadotrophin releasing hormone. This causes the pituitary gland to release 2 hormones. One of them is called follicle stimulating hormone, abbreviated FSH, and the other is called luteinizing hormone, abbreviated LH. Right. Now, follicle stimulating hormone, as you might expect, it stimulates the follicles in the ovaries to develop. Follicle stimulating hormone is also produced by men, and that hormone encourages the spermatozoa to develop. But in women, follicle stimulating hormone basically causes the follicles and the developing ova that are inside them to develop. Okay, now, so the brain produces a hormone called gonadotrophin release hormone, which causes follicle stimulating hormone and luteinizing hormone to be released. This happens during the first 2 weeks of the menstrual cycle, and the first 2 weeks of the menstrual. So a woman has her period, then everything is fine. The blood disappears and she starts over again. The first 2 weeks, the brain is releasing gonadotrophin releasing hormone, which is causing follicle stimulating hormone and luteinizing hormone to be released. This first 2 weeks of the menstrual cycle is referred to as the follicular phase. Okay, next. around 2 weeks into the menstrual cycle, there will be a spike or a sudden increase in FSH and LH that are released from the pituitary gland. And this sudden increase, this sudden spike of these two hormones causes the most mature ovum to complete its growth and be released. Right? So it is released, the most mature ovum is released from its follicle, and what's left of the follicle swells up into a structure that is called a corpus luteum. The corpus luteum releases other hormones. Two of them are called estradiol and progesterone. And estradiol and progesterone prevent the release of gonadotrophin release hormone, which in turn prevents the maturation of other follicles. So the follicular phase of menstruation, of the menstrual cycle, is characterized by the development of the eggs, basically. And then as soon as one of the eggs is released, what's left of the follicle swells up into a corpus luteum, which releases estradiol and progesterone, which prevents the other eggs from maturing because it temporarily stops the brain from producing or releasing gonadotrophin release hormone. So we go through 2 weeks where the eggs are developing, not all at the same rate, but one of them is going to mature first, and then when that one egg is released, it will essentially stop all the other eggs from developing. That second half, the second 2 weeks of the menstrual cycle, is referred to as the luteal phase. Right, so I could very well on the final exam ask you to tell me the difference between the follicular phase and the luteal phase of the menstrual cycle. Right, that could be a good exam, a good essay question, for instance. Okay, so here you see we have an ovary, and within the ovary we have lots of eggs in the process of developing within their follicles. Let's just take a close-up look at one of them. So here we have a primary oocyte developing inside a follicle. It grows and grows and grows until it becomes mature, and then it bursts its way out. So we have one egg being released. What's left of the follicle, the nurse cells and what's left of the follicle, swell up into a large yellow structure called a corpus luteum. The corpus luteum produces progesterone and estradiol, which prevents the other eggs from maturing. Eventually, over a period of about 2 weeks, the corpus luteum will kind of dry out and deteriorate. It will atrophy, as they call. Atrophy means wither away. It'll wither away. It'll become smaller, and then we start over again with the follicular phase. So this is a graph that's in your textbook. It's not very clear, but I'll do the best I can. So here we have the first 2 weeks of the menstrual cycle, the follicular phase, where if you draw a graph of the levels of follicle stimulating hormone and luteinizing hormone that are released into the body, you see that the first 2 weeks we have moderate levels of FSH and LH being released, and then we have a sudden increase or a spike. That sudden increase causes one of the follicles to release its egg, typically the one that's the most mature, and then what's left of the follicle swells up into a corpus luteum. The corpus luteum is producing estradiol and progesterone, which prevent the brain from releasing gonadotropin release hormone, so that it prevents the other eggs from developing. So progesterone, for instance, one of its jobs is to prevent the other eggs from developing. I'm going to show you in a minute that one of the birth control methods for women is to take hormonal birth control pills. I'm sure you've heard of this. What do you think is in those birth control pills? Artificial progesterone. So the artificial progesterone that a woman takes when she takes birth control pills is it is mimicking the effects of progesterone, so that it prevents. It basically fools the body into thinking that there's a mature ovary that has been a mature ovum that has been released into the fallopian tube, and so we've got to prevent all of the other eggs from developing until that one egg either gets fertilized or released. Basically, so what it does, progesterone, what birth control pills do is they fool the body into thinking that we're in a permanent permanent luteal phase of menstruation. This is just a close-up of a similar graph. So the brain releases gonadotrophin-releasing hormone, and then the pituitary gland, the anterior part of the pituitary gland, produces FSH and LH. They're produced during the first 2 weeks of menstruation during the follicular phase. There's a sudden increase or a spike, which causes one of the follicles to release its egg. Once the egg is released, what's left of it swells up into a corpus luteum, which produces progesterone and estradiol, which prevents the other eggs from maturing until another 2 weeks later, and then menstruation starts over again. Okay, so this is just showing that the corpus luteum, so here we have the follicles on the top, showing you what happens to this particular follicle after it releases its egg, swells up into a corpus luteum. The corpus luteum produces lots of estrogen and progesterone, and those estrogen and estradiol and progesterone cause swelling of the endometrium. So during the luteal phase of the menstrual cycle, the walls of the uterus, the endometrium, are swelling up and filling up with newly created blood vessels in anticipation of having to provide nutrients to a developing baby. If that doesn't happen, it's all released, as you see here. So that's the menses, the menstrual flow, when all of this tissue along with a lot of blood will come out because it's not going to be used. Okay, so there's the diagram. Okay, so once again, the brain releases gonadotrophin releasing hormone, which causes the anterior pituitary gland to release these two hormones. Those two hormones cause the follicles, the eggs inside the follicles, to mature. Eventually one of them matures the fastest and bursts its way out. What's left becomes the corpus luteum. What's left of the follicle becomes the corpus luteum that produces the estradiol and progesterone. And progesterone is what prevents the other follicles from maturing until the menstrual cycle is over. So in hormonal birth control pills, a woman takes fake progesterone, which fools the body into thinking that she's constantly in a constant state of luteal phase. Okay, now these hormones begin to falter in the late 40s or so, when a woman goes through a phase called perimenopause. So during the perimenopausal phase, which happens in the late 40s, hormone production declines and the menstrual cycle becomes irregular. There are other changes in the body, including hot flashes. Suddenly a woman feels very hot for no apparent reason. And this is just because some of the hormones are causing this sensation of heat, it's not a sensation, it's a genuine increase in body temperature, but it's caused by kind of an odd mixture of hormones, right? And typically during perimenopause, one of two things can happen to a woman's sexual desire. Some women start to lose their sexual desire during perimenopause. Other women, it starts to increase, but it generally doesn't stay the same as it was. Okay, then in your 50s, early 50s, you generally hit something called menopause, which is where ovulation stops entirely. So during perimenopause, you still have menstruation, but it's at irregular periods. It's usually around 28 days. A woman starts to menstruate typically when she hits puberty for the first time, around age 13 or something like that. And the first 1 or 2 menstrual cycles may not be regularly spaced apart. But then, from age 18 through age 45 or thereabouts, the menstrual cycle is pretty consistently 28 days or very slightly. And then when you hit perimenopause, it could be 2 weeks or it could be 6 weeks. Suddenly it's not 4 weeks anymore, but that's part of perimenopause. Okay, and then eventually the hormone production drops to the point where ovulation is the production of eggs, so ovulation stops, and there's a decline of estrogens produced by the ovaries, which also cause a decline in what are called female secondary sexual characteristics, which means that typically there's something called male and female secondary sexual characteristics, and these are physical traits about the body that identify you as a male or a female, right? So typically a young man has quite a different shape than a young woman. Man in his 20s has quite a different body shape than a woman in her 20s. As both men and women age, they have a reduction in secondary sexual characteristics because those secondary sexual characteristics were caused by hormones in the first place. So so this is typically in the case of women, that means certain things happen, like the voice goes down, so the voice is not as high pitched as a younger woman. The voice starts to loosen up and sound a little bit more like a man. Men at the same age, the opposite is happening. The lower voice is starting to go up. The women can start to develop facial hair. All women have a little bit of facial hair because there are hair follicles on the female face just like the male face, but they don't produce thick hairs the way the male does. But unfortunately for women, menopausal women, they start to sprout whiskers that you have to pluck out and things like that. And then eventually you become what's called a post-menopausal woman. Post-menopausal women are, in their 60s and so on. And women tend to dread this phase, and men do as well. Nobody likes to get older, but once you're on the other side of it, a lot of women say they enjoy being post-menopausal because they're kind of out of the game, and they can just appreciate life for itself. You have quite a shift in attitudes for many postmenopausal women. All right, so the sex hormones. There are sex hormones we mentioned briefly during the endocrine. Talking about the endocrine phase, there are sex hormones that cause the male and female body shapes to develop differently, and those sex hormones are they're produced in adolescence. They are produced in the gonads, and they give typically the male and female body shape and the male and female secondary sexual characteristics. Okay, in the case of men, testosterone is an example. Right, so testosterone is a hormone that causes men to develop into the shape of typical men. Shoulders are wider than the hips. Men can develop muscle more easily. They can lose fat more easily. They can, develop more muscle mass if they exercise and so on. Testosterone makes you hairy on the body, but it also causes the hair to fall out of your head. So typically, if you see a man who goes bald, part of that is genetic, part of it is caused by hormones, and a man who has an excess of hormone will tend to go bald faster than a man with the same genetic makeup who produces less testosterone, for instance. In the female body, the estrogens are a class of hormones that lead to the female secondary sexual characteristics, the kind of hourglass figure, development of breasts, lack of body hair as opposed to men developing body hair. So these are examples of secondary sexual characteristics. So you notice, of course, that before adolescence, boys and girls, they have different genitals, of course, they have different sex organs, but the body shape is basically the same between a boy and a girl until they hit adolescence, and then the body shapes begin to diverge. So secondary sexual characteristics, biologists define secondary sexual characteristics as physical features about the body that identify you to the opposite sex and which identify you as either a male or a female, but which are not directly involved with reproduction. They're not directly involved with the reproductive act. They're simply signals or signs that you're a male or a female. So these things happen in the animal kingdom as well. So for instance, in the animal, with deers, the males develop these antlers, and that's a male secondary characteristic that females don't get. Finches, one of my favorite birds, the males have secondary sexual characteristics. They have these orange patches on their cheeks, whereas the female does not. In general, in the bird world, the male is usually more elaborately decorated than the female, and his job is to attract a female. So here we have some peacocks. So this is a male peacock that's acting very flashy to try and attract a female, right? So humans have secondary sexual characteristics like that as well. This is actually not one of them, but a lot of middle-aged men think that this kind of thing, buying one of these, makes you more attractive. Really, it just makes you look silly. So this is not officially a male secondary sexual characteristics, but human males try to compensate in other ways. Now, one of the interesting things about secondary sexual characteristics is that they make you attractive to the opposite sex, but the interesting thing is that many of these secondary sexual characteristics disappear after the typical age of mating. So the typical age of reproduction in humans is around 20 years. So the secondary sexual characteristics that attract a mate begin to increase in adolescence. They hit their maximum in the 20s, and then they start to decline. But I guess as far as nature is concerned, as far as evolution is concerned, it doesn't matter if you lose all of your secondary sexual characteristics at age 40 because you've already had all the children you're going to have, or something like that. So you will notice that secondary sexual characteristics begin to decline in older age. Here's, for example, here's Prince William when he's about 17 or something, I think, and then here he is when he's in his early 20s. The hair is just, the women were attracted to the full head of hair, which basically disappeared very quickly. So that's the same for both male and female secondary sexual characteristics. It's an interesting thing from an evolutionary perspective that the things that are used, the signs that are not necessarily related to reproduction, but these signs that are used to attract a mate begin to decline after when they're no longer needed, essentially. All right. Now, what I've talked about so far is conception. Conception means to conceive or to produce children. And at some point, we decided that, humans decided that we wanted to be able to have sex without producing children. And so we came up with the term contraception. The word contra means against. So again, it's kind of a play on words. So we came up with the word contraception, which is commonly called birth control. So I'm going to explain how some of the birth control methods work. Some of them you've already heard about. Okay, so first of all, the birth control methods are grouped into several different groups. One group is called barrier methods, where basically you put a physical barrier between the sperm and the egg. Example, there is a thing called a condom, which is basically a rubber sheath that fits over the penis and contains the sperm. So when the sperm comes out, it doesn't reach any of the eggs. It never makes it into the uterus or in the fallopian tubes. There is a male condom, which is the most common, but there is a more expensive but equally effective female condom. So in the male condom, the rubber sheath fits over the penis. In the female condom, there's a ring, a wire that fits outside the vagina, and then the sheath gets pushed into the vagina, so that the sheath will catch the sperm and prevent it from getting up inside the cervix and the uterus and so on. If these are properly used, they are about 98% effective in preventing conception, in preventing pregnancy. Those are the barrier methods. Okay, next we have surgical methods. So you already heard about the vasectomy, which is where the male gets his two vas deferens cut, so the sperm can't even get to the penis. That's 99% effective. The only time it's not effective is if the surgeon makes a mistake and fails to connect or tie the vas deferens tube shut. The female equivalent of a vasectomy is called a tubal ligation, which is where the fallopian tubes, the oviducts, the fallopian tubes are cut and then tied in a knot to prevent the eggs from reaching the uterus. Both of these are 99% effective, and both of them have a moderate chance of being reversed if you change your mind later. So if you have a vasectomy or a tubal ligation because you're determined you don't want to have children, and then later you change your mind, a surgeon can possibly connect it. I haven't seen can possibly reconnect you again. I haven't seen the statistics recently, but last time I saw the reversal rate for vasectomies were something like 70 or 80% successful. So if you get disconnected because of a vasectomy, there's a 70 or 80% chance that a good surgeon could reconnect you, and then you could have children again. I believe the rate for successful reversal of tubal ligations is a little bit less than that. Okay, now the hormonal methods, we heard about those already. Birth control pills is where you take pills that contain artificial progesterone, which fools the body into thinking that it's in the luteal phase permanently, basically. These are only about 92% effective. A woman has to take a pill every single day in order to make this work. If she skips a day or two, she could become pregnant quite easily because the hormones, the progesterone levels may drop to the point where an egg could be released. So the hormonal birth control method is not that effective. It's kind of a nuisance because you have to take the pill every day. It's only 92% effective when it works, and it has dangerous side effects on the woman's health, including high blood pressure. So especially even higher blood pressure if the woman smokes. So it's dangerous. It is a bit dangerous, but nevertheless it is common because well, let's be honest, it's because the male doesn't have to do anything, and the female, the woman, is taking responsibility for birth control entirely on herself. I have to admit that's never struck me as being fair, that all of the responsibility for maintaining the reproductive health and conception and contraception and reproductive health of society is being borne by heterosexual women, which is definitely not fair. Men need to do their part as well. But anyway, so birth control pills. There is a method. I've written implants here because there is a method that they're experimenting with now, where they implant the progesterone under, they make progesterone in a pill form. Basically, they implant pills or little bits of progesterone under the skin of the woman every few months, so she doesn't have to go through the trouble of taking pills every day, because sometimes if you forget to take a pill, you could get into big trouble that way. All right, now there are some behavioral birth control methods. One is called the withdrawal method, which is withdrawal method is the common name for, the technical name for this is called coitus interruptus. The word coitus is the technical word for mating, for having sex. Interruptus means to interrupt it before it's finished, which means that the male pulls out before the sperm comes out. This is called the withdrawal method, coitus interruptus. If it's properly used, it's 70% effective, which is not particularly effective. That means if it's done properly, it only fails to prevent pregnancy 7 out of 10 times. So that's a lot of chances, and furthermore, it's rarely used properly. It is rarely done properly because, as some of you may know, the man doesn't want to pull out until before he's finished. He doesn't want to do that. So he may not. So coitus interruptus, not particularly effective, and very rarely is it properly used. All right, now here's an interesting one called the rhythm method. Now, as you know, Catholicism is the oldest form of Christianity, and so Catholics and other Christians believe that technically their belief is that God intended for sex to be a reward for having children, and therefore you shouldn't be trying to short circuit that. You should only be basically using sex to have children, not just for fun, not just for pleasure. That's considered to be a kind of a sordid, selfish thing to do. So, but nevertheless, people, Catholics included, really did want to have sex, and so they kind of pressured the Catholic Church to figure out a way that they could have sex that was in keeping with that idea. And so what the Catholic Church came up with was the idea that yes, you can have sex as long as you don't do anything as long as you're not doing anything to prevent, as long as you're not doing anything that stands in the way of becoming pregnant. So this obviously is only because it's a religious organization. This advice is only for married people. The Catholic Church and all the other Christian sects believe that people should not have sex unless they're married, and then if they do have sex, technically they should only be doing it with the idea of having children. But what the rhythm method is it was a kind of a compromise position that the Catholic Church came up with, where they said that it's okay you can have sex during the first 11 days of the menstrual cycle, because that's the follicular phase where there is no egg that has broken out of no egg has broken out of its follicle yet, right? So the odds of a woman, if during the first 11 days of the menstrual cycle, if a woman has sex, the odds are that she will probably not get pregnant because an egg has not been released yet. During the luteal phase, the odds are very high that she will get pregnant. So then this rhythm method means that a woman keeps very close track of her menstrual cycle, and she and her husband only have sex during the first 11 days of the menstrual cycle. And when this is properly used, it's 90% effective, which is actually quite impressive for something that actually doesn't do anything to separate eggs and sperm, right? So 90% effectiveness for the rhythm method is actually quite impressive. All right, now, as young people who are listening to this lecture, probably the one thing that you're hearing from older people is the word abstinence. The word abstinence means simply not to have sex, right? So the most certain way to not get pregnant is to simply not have sex. And so that is true. Abstinence is the most effective birth control method, the most effective method of contraception. I know, however, that that's something that's easy for somebody who's 50 to say. It's something that's very difficult for somebody who's 20 to actually do. And I remember that, so I'm very careful about giving that advice. But it is true that it is the most effective way. All right, let's move on now to the other downside of sexual relations, which is that things called sexually transmitted diseases, or STDs. Okay, so let's discuss some of the different types of STDs. Okay, so some forms of sexually transmitted diseases are caused by bacteria, and bacteria can be easily killed by special types of drugs called antibiotics. Right? So one of them is called syphilis. It's caused by a bacteria called Treponema pallidum. I do not expect you to remember that. And if it's not checked, it can cause birth defects in the developing child. There's another one called gonorrhea, and there's another one called chlamydia. Syphilis, gonorrhea, and chlamydia are all caused by bacteria that are simply transmitted from a man to a woman or a woman to a man during sex if one of them already has it. Okay, but as I said, it's relatively easy to treat these diseases with antibiotics, because antibiotics are drugs that specifically kill bacteria. There is one sexually transmitted disease that's caused by a protozoa. It's called trichomoniasis, which is caused by a protozoa called Trichomonas vaginalis. It can be treated by a drug called tinidazole. And now we get into the viral sexually transmitted diseases. Viruses can only be prevented by vaccines. They can't necessarily be cured by treatments. So you might have heard in the news in the last year or so, when we've been talking about COVID, we've been talking about COVID vaccines, and of course a vaccine is used to train your immune system to kill a virus before it can infect you. That is a totally different thing than viral disease treatments. Right? So in the world of trying to fight COVID-19, for instance, we have COVID-19 vaccines that ideally prevent you from getting COVID. But once you get COVID, we have certain treatments. So there are certain treatments that you can use once you've caught a disease. Well, there are three very serious sexually transmitted diseases that are caused by viruses. One of them is called herpes genitalis, which is caused by a virus called human herpes virus 2, HHV2. There's another one called genital warts, the technical term for warts is called a papilloma, and so those genital warts, this is ugly warts on the genitalia. This is caused by a family of viruses that are called the human papillomaviruses, or HPV. Now, some of these viruses just cause ugly, unsightly genital warts, like HPV-6, but some of them cause warts and cervical cancer. So you know what the cervix is. It's the entry to the vagina. And if you get HPV-16, you could actually get cervical cancer. There is a strong connection between catching HPV-16 and cervical cancer. Finally, probably the worst one is AIDS, the human immunodeficiency virus. HIV stands for human immunodeficiency virus, which is a virus that attacks and kills T cells, and so thus it destroys the immune system. Now, herpes, we have never developed a vaccine to herpes. Never been able to do that. We have never been able to successfully develop a vaccine to HIV. But we have developed a very successful, effective vaccine that prevents getting infected by HPV-16 and thus prevents women from getting cervical cancer. Okay, so of the three main venereal diseases, sexually transmitted diseases, or STDs, HPV-16 is the one that we have the most effective vaccine against. So you would think, well, we should be giving this vaccine to all of the girls in the world before they become sexually active. Of course, many of them are going to remain. Many of them will practice sexual abstinence. Many of them will wait until they get married, whatever. But why not just vaccinate all the girls against HPV-16 so they won't get cervical cancer? Well, unfortunately, there was a movement to vaccinate all the girls against HPV-16 and thus against cervical cancer. But there is, as many of you have noticed, there is a very strong anti-vaccination movement in the world where people think that vaccines are evil and so on. And there is a very strong sect of people who resist the idea of vaccinating teenage girls against HPV-16 because they think that will make them promiscuous. They think that it would be better off to have them be scared of catching HPV-16 and thus being scared into being abstinent than it is to give them protection from HPV-16 and then think, well, now I'm invincible, so I will have sex with every man that I see because I'm not afraid of HPV-16 anymore. I don't think that'll happen. You know, I think if you want to if you're intent on teaching your daughters about morality, you can do that, and you can also vaccinate them at the same time and say, you know, there's a saying in English where they say, be good, but if you can't be good, be careful, right? That's essentially what that means. It means, you know what the right thing and the wrong thing to do is, but I understand you're not immortal, you know, you're not invincible or infallible or something, so be good. If you can't be good, be careful. All right, let's talk for a minute about which of these contraception methods is actually effective at preventing STDs. Okay, so which do you think? Do you think the barrier method would be effective? What about the surgical method and the hormonal methods and the behavioral methods? Okay, so in fact, only two of these methods on the list, only two of these contraceptive methods are equally effective at preventing sexually transmitted diseases. The barrier method, which uses male or female condoms, is quite good, but not 100% effective, but somewhat effective pretty good at preventing sexually transmitted diseases from being transferred. And of course, abstinence. If you don't have sex, you won't get sexually transmitted diseases. Okay, a vasectomy and a tubal ligation is useless at preventing sexually transmitted diseases. Hormonal doesn't do anything. Hormonal birth control pills doesn't do anything to prevent catching sexually transmitted diseases. The withdrawal method, not really, because most of the agents, the microbes that cause the sexually transmitted disease, are present on the skin, not necessarily in the sperm or in the vaginal secretions. So coitus interruptus is not particularly good. The rhythm method isn't either. So really the only two methods for preventing transmission of sexually transmitted diseases are condoms and abstinence. If people are married or whatever, or they're in a committed relationship and they're convinced that they're never going to have sex with anybody except this other person for the rest of your life, you're both tested for sexually transmitted diseases, and if you don't have them, then you just have sex with that person only and you never have to worry about it. So that's another thing. Now let's move on to the topic of human gestation. Gestation refers to pregnancy and birth. So we mentioned the fact that fertilization takes place in the oviduct, and you have the possibility of an ectopic pregnancy, right? So the zygote continues to divide, turns into a blastula, implants itself in the uterine wall, and develops a trophoblast, which later becomes the placenta. And then the blastula kind of curves in on itself and becomes a gastrula, and thus when it turns in on itself and becomes a gastrula, it actually creates 3 different distinct layers of tissue. All right, so here we have the eggs maturing in the ovaries and traveling down the oviduct. Fertilization generally happens about there, just as the egg is leaving the ovary. Then it implants itself on the uterine wall and creates a trophoblast. The trophoblast will become the placenta, which allows nutrients and oxygen to be exchanged between mother and child. So here we have a close-up of the blood vessels inside the placenta. Notice that the mother's blood and the baby's blood are not actually touching. It's just that they come close enough together that these nutrients and oxygen can cross the thin layer of tissue that separates them. All right, now typically the female gestation, the pregnancy period, is 9 months in length, and that 9 months is characterized by 3 very different trimesters, so that are 3 months each. Okay, we'll talk about that later. Okay, so gastrulation, I mentioned, is where the blastula bends inwards to form 3 layers. So when this soccer ball bends inward, you have an outer layer, which is called the ectoderm. You have an inner layer, which is called the endoderm. Remember that generally in Latin, endo means inside, exo means out, or ecto as well. So endoderm becomes the inner layer, and then the mesoderm, meso means the middle, so the mesoderm forms the middle layer. The endoderm of the gastrula develops into the lungs and the digestive system. The ectoderm, the outside, develops into the skin and the nervous system, and the mesoderm, the middle layer, becomes the muscles. That's the only the only study of gastrulation that I expect you to know. Is just that the endoderm becomes the lungs and the digestive system, the mesoderm becomes the muscles, and the ectoderm becomes the skin and the nervous system. So all of these tissues in an adult human are developed from these tissues in a developing gastrula. Now, during development, the developing child has to develop organs, and obviously that happens after the gastrula phase. So generally, when the developing child is developing organs, this happens during the first trimester. In fact, during the first month and a half, the organs develop, and that phase is referred to as organogenesis. And during organogenesis, the developing child is particularly susceptible to damage and mutations and birth defects. So anything that a developing embryo can be exposed to that results in a deformity, in malformation or deformity, is referred to as a teratogen. A teratogen is something that causes mutations or birth defects. All right, so here we have our blastula. Right, the blastula curves inward and forms an endoderm, an ectoderm, and a mesoderm, and those 3 layers of tissue develop into different things in the adult. The study, as I said before, the study of development is called embryogenesis, or embryology rather. So there are scientists and even doctors that study the development, human development, that call themselves embryologists. Okay, so this is just a more detailed map of the tissues that the endoderm, mesoderm, and ectoderm turn into. I don't expect you to remember them all, just the ones I talked about earlier. Okay, so we have our 3 trimesters. In the first trimester, you have embryogenesis and the production of an embryo. First trimester, you have organogenesis, development of the organs, and the blastula develops into an embryo. Second trimester, you have a fetus. Third trimester, you have a very well-developed fetus, which then comes out, and we rename it as a neonate. Okay, so just remember that organogenesis occurs in the first trimester. Now what happens when we're trying to deliver the baby? The baby wants to come out after 9 months. Right? There are actually 3 stages of delivery. During the first stage of birth, the first stage of delivery, the cervix dilates, it opens up. During the second stage, the smooth muscles that surround the uterus contract to push the baby out as a result of the hormone oxytocin being produced in the brain, in the pituitary gland. Right, so we talked about that during the endocrine lecture. Right? And if so during the period where the cervix is dilating but the baby hasn't pushed its head out yet, that's what we commonly refer to as labor. And labor, the amount of time that a woman is in labor, and labor is very unpleasant because you have these contractions and you have pains and it hurts, and the vagina doesn't really want to stretch that much, doesn't really want to, you've seen the size of a baby's head, you've seen the size of a vagina, you know that that's going to hurt, right? So it's not very pleasant, but you can, if you get tired of it and you want to just get the baby out and get it over with, you can induce synthetic birth. You can force the uterus to push the baby out by giving it an artificial version of oxytocin called Pitocin. Right? So if you've had enough with the labor, get a shot of Pitocin and let's get the baby out. Stage 3, after the baby has come out, the placenta has to be delivered sometime later. The amount of time that it takes for the placenta to follow the baby is variable, but it's not nearly as difficult. The hardest part of the birth is getting the head out. Once the head is out, the body comes out relatively easily, and the placenta comes out later very easily as well. If you run into problems, the baby typically has to turn upside down so its head is facing downwards to come out. Sometimes the umbilical cord that that feeds the baby oxygen to the placenta, sometimes the umbilical cord gets wrapped around the baby's neck or something, for instance. You need to get the baby out quickly. You have no choice but to do something called a cesarean section, which is surgery where you cut the baby out. So that's what a C-section is. That would leave you with a permanent scar usually, and it's usually a bit dangerous. So they avoid using a C-section, a cesarean section, but sometimes they have to. Okay, so the baby turns upside down just before birth. You can see the umbilical cord here. The umbilical cord goes into the umbilicus, the navel, the belly button, and it's attached on the other end to the placenta, which is bringing the mother's and the baby's blood vessels into close contact so that nutrients can be exchanged. So the 3 stages of birth, we have dilation of the cervix, right? So that this opens up enough to let the baby out. The baby's head comes out. When the top of the head is coming out, they say that it's crowning. And then after the baby has come out, the placenta is delivered later on. The placenta has to come out as well. Now, because human evolution is funny, the human head has been getting bigger and bigger over the millions of years that we've been evolving, but the human vagina has not. So we're in this situation where it is actually quite dangerous today for a woman to give birth. There are a lot of things that can go wrong. So there are a lot of things that can go wrong with birth. Usually they don't, but they can. And one of the problems that's more common than you'd like it to be is the head not being able to come out through the vagina because it's not big enough. In that case, the doctor may do something called an episiotomy in 12% of the cases, which is where they take a pair of scissors and they cut the vagina in order to let the baby's head out. This is one of the reasons why we celebrate our mothers on Mother's Day. So if you see your mother, thank your mother for giving birth to you, because she probably went through a terrible ordeal and risked a great deal by giving birth to you. So it's not, giving birth is not a trivial thing. So in this case, we have an episiotomy, which is kind of a scary thing to think about, that in 12% of the cases of birth, they'll actually have to, the doctor will actually have to cut the vagina to let the baby's head out. Okay, now, as if that wasn't hard enough, in about 15% of the cases, women who've just given birth, about 15% of the time, suffer from something called postpartum depression. Partum means giving birth, and post means afterwards. So after birth, they suffer from postpartum depression, PPD, about 15% of the time, where they get very depressed and anxious about life. So that's another thing that mothers have to cope with, an all too often occurrence, to get very anxious and very depressed about the future and things like that. Now, let's talk for a minute about things that can go wrong during birth. I mentioned earlier that a teratogen is a chemical agent or a biological agent, and by biological agent I mean a virus or a bacteria or something like that, that can cause birth defects. Now, the word birth defect is not very politically correct, but we haven't thought of a better word yet. So it means that something goes wrong with the baby. Okay, so a teratogen, we call something that can cause a birth defect a teratogen. Example, fetal alcohol syndrome is a condition that some babies will have if their mother drank too much or drank a lot of alcohol during pregnancy, which is a very sad situation. There could also be genetic accidents. So I already mentioned, Turner syndrome, Klinefelter syndrome. Those things are not terribly serious, but there are other ones that are genetic accidents, like Down syndrome, for instance. So as I mentioned before, if you have 3 chromosomes instead of 2 for a particular chromosome set, for example, there are 23 chromosomes in the human genome. If you have 3 copies of chromosome 21 instead of the 2 that you're supposed to have, you will have something called Down syndrome. Down syndrome. So you've probably heard of that. And finally, you could inherit genetic conditions that are caused by single gene mutations, mutations to single genes. Things like cystic fibrosis, muscular dystrophy, a lot of those birth defects are caused by mutations to genes that the child inherited from their parents. Let's look at some examples of teratogens. So there is a drug, historically, this is kind of interesting, there was a drug called thalidomide, which is a sedative which calms you down when you're anxious. And that drug was not properly tested by the pharmaceutical companies when it was invented. And it turned out that if pregnant women took thalidomide, their children would be born without arms and legs. This happened to thousands of children in between about 1960 and 61. I was born in 1961, and so I actually went to university with a guy who had thalidomide syndrome, and he had no arms, but he did have legs and feet, so he was lucky in that sense. And he used to drive to Simon Fraser University using his feet. Operate the steering wheel with his feet. He wore sandals, so he'd walk up to a door and kick off one of his sandals, and then open the door with his foot, and then put the sandal back on. So there are people who had these kind of birth defects. So thalidomide is a chemical that turned out to be a perfect example of a teratogen. Alcohol is another chemical that's a teratogen. It causes something called fetal alcohol syndrome. Rubella is the virus that causes German measles. There is a vaccine against it, and most people in the Western world get vaccinated against rubella in the first year of life. typically children in the Western world get what's called the MMR vaccine, which stands for measles, mumps, rubella, and then it's often also called the MMRV vaccine, stands for measles, mumps, rubella, varicella zoster. And so those are 4 viruses that can cause problems. So most people in the Western world are vaccinated against rubella. So having rubella during pregnancy can cause birth defects, but it's usually not a problem because most women were vaccinated against rubella when they were 1 year old. So there's no danger of you, if you're a woman, there's no danger of you catching rubella during pregnancy. If you haven't been vaccinated against rubella, you should be, because if you're a woman and you get pregnant, it could cause birth defects in the baby. In fact, it will cause something called congenital rubella syndrome. Okay, syphilis is a sexually transmitted disease caused by a bacteria called Treponema pallidum. It can cause bone defects and birth defects. It used to be a common cause of birth defects in the Middle Ages in Europe, where a woman would get syphilis, usually from a man, obviously from a man, but if she got pregnant and she had syphilis, the baby would be born with deformities, bone deformities. Toxoplasmosis is something that's still a threat today. It's caused by a protist called Toxoplasma gondii. You can catch it from cat feces. It is carried by domestic house cats. So if you're pregnant, you should not be changing the cat's litter box, or you probably shouldn't be handling the undercooked meat. Let somebody else do that. Let somebody else change the cat's litter box, but stay out of contact with cat feces, cat excrement. Okay, so these are 5 different teratogens. 2 of them are chemical teratogens, and 3 of these listed here are biological teratogens. So on the final exam, I could ask you about biological or chemical teratogens. Okay, so here's a poor child that had thalidomide syndrome. Notice that he's born with no arms and legs, but he does have hands, fingers, and feet that are just on the end of stumps. Here's a baby that unfortunately is born with congenital rubella syndrome. So his mother had German measles during pregnancy. Congenital rubella syndrome, the symptoms include microcephaly, which means a small head. Cephalic, of course, means the head. Problems with the heart and cataracts on the eyes. It's very unpleasant. The microencephaly, meaning a small head, is accompanied by mental retardation, so it's bad. So it's much more simple just to get vaccinated against rubella to make sure that you never catch rubella during pregnancy. Okay, fetal alcohol syndrome is caused by drinking too much during pregnancy. There's no way that you can tell except by characteristics of the baby's face. Typically, one of the things that a fetal alcohol syndrome child will generally have a short temper and maybe a short attention span, have trouble learning. It's quite sad. One of the signs that you can look for is an indistinct philtrum. You know, that little crease that's under your nose? That's called the philtrum. And if that is shallow or missing, that's a clue that maybe that person had fetal alcohol syndrome when they were born. Okay, here we have our friend the Hunchback of Notre Dame. The Hunchback of Notre Dame was a legendary Middle Ages monster, as it were, and he had bone deformities. The original story was that, we have this is a Disney story which is meant to be happy, but the original story was meant to be sad and horrific. That Quasimodo was his name. Modo means man, quasi means almost, so Quasimodo means subhuman, basically. So they're talking about him as subhuman. And he had this hump on his back because his mother had syphilis. It was a way of saying that his mother was an unvirtuous woman. So if you had these kind of bone deformities, people would shun you and treat you like an outcast because it meant that your mother was not virtuous. Although that's not your fault, but people had very funny attitudes in those days. So Quasimodo, the Hunchback of Notre Dame, they were suggesting that he had a hunchback because his mother was impure. Toxoplasmosis, you can get it by domestic house cats, particularly by handling the feces. So pregnant women should refrain from handling house cats or changing the litter box. All right, what about other genetic accidents? Okay, we already talked about triploid and trisomy and monosomy. Those are examples of something called aneuploidy. Aneuploidy means you have an odd number of chromosomes when you're supposed to have an even number. So we're supposed to have 2 of every chromosome. We should have an even number of chromosomes. Aneuploidy means we have an odd number because we have 1 too many or 1 too few of one set. Okay, this happens due to a genetic accident during meiosis, which is called non-disjunction. Don't worry about it too much, except that non-disjunction means that 2 chromosomes are supposed to separate and go independently into 2 different sperm cells or 2 different egg cells. But instead they got stuck together so that both of them went into one egg and neither of them went into the other egg. And therefore you end up with one egg that has 1 too many and one egg that has 1 too few chromosomes. If you then pair either of those trisomic or monosomic, or you pair either of those malfunctioning eggs with a normal sperm or vice versa, then you end up with aneuploidy. So, for example, we already talked about Turner syndrome. Turner syndrome is where you have one X and nothing else. So you say that it is somebody that has Turner syndrome is monosomic for the X. Down syndrome is where you have 3 of chromosome 21, and so then you say that is trisomy 21. You say somebody is trisomic for the 21st chromosome. So this is called a karyotype. This is a picture. You get some white blood cells and you extract the chromosomes from them, and then you put them onto a microscope slide and you take a picture, and then you cut the picture up and you put together all the paired chromosomes so that you can tell whether somebody has a normal set of chromosomes. That's called a karyotype. So here we have a karyotype, and notice that this person has 2 of every chromosome except number 21, where they have 3 chromosome 21s. So this is trisomy 21 or Down syndrome. Okay, now look at this karyotype for 1 second and tell me whether this is a boy or a girl, whether this is a man or a woman. Obviously this is a woman's karyotype because they have 2 X's. Okay, so the symptoms of trisomy 21 or Down syndrome are short stature, meaning you're short, physically short. You have an epicanthal fold of the eye, which is makes your eye look kind of Asian. You have a broad face, a very large tongue, so that people who have Down syndrome often kind of walk around with their tongue kind of sticking out because it's more comfortable. It's a very large tongue, so it's kind of uncomfortable to keep it in your mouth. Okay, and then some heart problems, mental retardation, and some digestive issues. There's a particular type of crease on the hand called a simian crease, and a shortened 5th finger. Right, so these are some of the symptoms of Down syndrome, trisomy 21. All right, now here's something that you should know if you're ever interested in having children, and that is that the odds of having trisomy 21, the odds of having Down syndrome increase with the age of the mother. So the age at which the woman becomes pregnant increase the chances of having Down syndrome, the baby having Down syndrome. So you see here that if you are 20 and you have a child, the odds of having a child with Down syndrome are 1 in 1,550. Right, so 1 in 1,500, that's quite low. Then the odds increase if you're having children in your 30s, and if you have children in your mid-40s, the odds of having a Down syndrome baby is 1 in 25. Right? That's a dramatic increase relative to when you had children when you're young. So that's something that you should be aware of if you're having children, because many women, in order to make room for the career, are putting off having children until later. But you should, knowledge that it's a terrible thing to think that you have to choose between career and children. I mean, it's totally unfair that men don't have to make that decision, and women do. But nevertheless, knowledge is power, and so you can always take better control of your life if you know things. So I'm just telling you that one of the downsides of waiting until later in life to have children is that the odds of having a child with Down syndrome increase exponentially with age. So you're safe until around 30. As soon as you start getting up into having children in your 30s and 40s, the odds of having a Down syndrome child increase quite a lot. And we already talked about the symptoms and signs of Turner syndrome. Okay, let's just touch briefly on some of the genetic mutations that can be passed on to a child from the parent. I will not ask you about which chromosome is infected in any of which chromosome is affected in any of these diseases, but be aware that cystic fibrosis it's a coughing disease. We talked about it earlier on, and cystic fibrosis. Um, Actually, I don't think I did cover cystic fibrosis, so forget about that one. But it is caused by a single mutation to a single gene, to just one gene, as opposed to like Down syndrome. Trisomy 21 is not a mutation to one gene; it's an imbalance of 1,000 genes, because you have three chromosome 21s when you should only have two. That means the dosage of every gene that's on chromosome 21 is higher. All right, now there's another genetic mutation called Tay-Sachs disease, which causes mental retardation. It's a brain issue. It's caused by a mutation in one gene on chromosome 15. Phenylketonuria is a disease that prevents you from eating certain types of amino acids. You can deal with that by altering your diet. Huntington's, formerly known as Huntington's chorea, is a very tragic disease, which is caused by a single mutation on chromosome 4, and it's very tragic because it's a dominant mutation, which means that we now have the ability to do a DNA test and tell whether you're carrying the mutant gene. And if you have the mutant gene, you're guaranteed to get Huntington's, and Huntington's is characterized by insanity, which means that if your father went insane and he was diagnosed with Huntington's, then you have to decide whether you want to go to the doctor and have a genetic test done to find out whether you have Huntington's or not. Would you want to know that? Well, you might want to know that. If you have Huntington, if your father had it, and then you have it, you're guaranteed to go insane when you're in your 40s, and that's not something that you would necessarily want to know. But you might want to find out if you were planning on having children with somebody, because you may or may not want to pass it on to them. So there are various reasons why you may not want to know, or you may want to find out, or you may not want to find out. But Huntington's is a particularly tragic disease. Muscular dystrophy is as well. It causes deterioration of the muscles. It's caused by a mutation to a gene on the X chromosome, which means that because women have two X chromosomes, if you have a mutant gene, which is what's known as a recessive mutation, you have two X chromosomes, the odds of showing the disease are less, because the other chromosome probably has the functional gene on it. So the fact that you have a dysfunctional gene on the other one, the other X chromosome, doesn't matter as much. Okay, but if you're a boy, you have one X, and if it has the mutation, you don't have another X to cover it, to mask it. You just have a Y. So there tend to be more boys in the world that have muscular dystrophy than girls. Hemophilia is the blood clotting disease. Your blood fails to clot. That, again, is caused by a mutation to a gene on the X chromosome. So I mentioned X chromosomes and what are known as X-linked genetic mutations. So this is where you have a mutation on the X chromosome. Most of these X chromosome mutations are what are called recessive genetic mutations, which means that they only show up if you have only one X chromosome. If you have two X chromosomes, you may have the mutation on one of the X chromosomes, but the other X chromosome, the gene on the other X chromosome is okay. So the disease never gets a chance to express itself because it's being hidden by the functional gene. But nevertheless, these X chromosome genetic mutations tend to show up more commonly in men than women because men only have one X chromosome, and so they don't have another X chromosome to hide it or to mask the mutation. So examples of X-linked genetic mutations include pattern baldness. That's what they mean when they say baldness comes from your mother's side of the family. So if you think about that, if you're male, you got your Y chromosome from your father, and therefore the only place you could have gotten your X chromosome with the baldness gene on it, with the baldness allele on it, you got it from your mother. Okay, color blindness as well, that's another one. Hemophilia, which we already mentioned, and muscular dystrophy. So these are 4 examples of X-linked genetic mutations or X-linked genetic diseases. So I could ask you about those on the final exam. Certainly if you go on and you take more biology and you take biology 234, which is the genetics course, you'll learn much more about this. Okay, now finally, we've done with the anatomy and physiology part. I'm just going to move into the social part of this and talk a little bit about things that are interesting to arts students who study philosophy and political science and history. And I'm going to talk about eugenics, which is a bizarre scientific movement that appeared in the 30s, and in the 30s was about the time that we started to realize how genes work. Right? So genetics became something that the average person, these days everyone knows what a gene is and sort of knows what genes are and what genetics is, but the average person didn't really know that prior to the 30s or the 20s. But as soon as they started to figure out that characteristics, both good characteristics and bad characteristics being things like muscular dystrophy and other genetic mutations and things that harm people, all of those things are carried on genes. They're examples of genes that are carried on chromosomes. So the idea of eugenics was a movement or a philosophy that sprang up in the 30s, where they said we can improve the human race by controlling who gets to reproduce. If we can stop people who carry these mutant genes from reproducing, we could actually rid the human race from these mutations. That was what the idea was. It didn't necessarily work out that way. Okay, so eugenics was a philosophy that espoused the elimination of birth defects from populations through selective reproduction, deciding who gets to reproduce and who doesn't get to reproduce. Started in the 1930s, right? Historically, it's had some very dramatic effects. So, for instance, in Nazi Germany, the Nazis involuntarily sterilized people who had birth defects, people who had Down syndrome and things like that. And then, of course, as they got more and more crazy with these Nazi and fascist ideas, they were looking into the idea of stopping cultures that they didn't like from reproducing. So they were looking into sterilizing Jews, for instance, and eventually ended up just murdering Jews instead. That's one example. Another example is the voluntary refraining from having children of people who have Tay-Sachs disease in their family tree. So that was a form of eugenics that took place voluntarily, which did a lot to actually eliminate the Tay-Sachs disease mutation from the population. Okay, so forced eugenics, example, the Nazis with the sterilization of people who had birth defects, and Tay-Sachs. It turned out that Tay-Sachs was a genetic mutation that was was very common among Ashkenazi Jews, which are, in other words, European Jews. And what they would do is if two people were thinking about getting married and they both had an example or examples of relatives that had Tay-Sachs disease, they would go to a doctor and say they would give the doctor their family tree, their pedigree as it's called, and the doctor would look at the two pedigrees and then calculate the odds that this couple would have a child that had Tay-Sachs disease. And if the odds were very high, the doctor would say, well, the odds of your having Tay-Sachs disease, if you two get married and have children, the odds of them having Tay-Sachs disease are very high. Do you want to think twice about maybe marrying each other? And if you do marry each other, maybe you want to think twice about having children? Maybe you'll just adopt children and not have any of your own. And so this practice was carried out by the Ashkenazi Jews for a couple of generations, and they succeeded by doing this completely voluntarily. They succeeded in practically eliminating the disease, the Tay-Sachs disease. This was actually a group of Ashkenazi Jews that migrated to North America. The frequency of Tay-Sachs disease in that population used to be very high, and that now it's very low. They almost completely eliminated it through voluntary behavior as opposed to political forcing behavior. Okay, but unfortunately, modern eugenics is still being practiced by some people because in the modern day, we can test what kind of genes our children have before they're born. You can do something called amniocentesis, where you puncture a needle into a pregnant woman's abdomen. You draw out some of the fluid that the baby is sitting inside the placenta, and you test the chromosomes. And so that fluid is called the amniotic fluid. And you can tell whether that child is carrying muscular dystrophy or Down syndrome or something like that. You can also tell whether it's a boy or a girl. And then, if you want, you can selectively abort children based on that. So I don't know how you feel about that. That's another discussion. But that is possible in the modern era, and it is being done in the modern era. So on the grounds of amniotic testing, people are selectively aborting girls because traditionally boys were, families, some people want boys more than girls, and so it does happen. This is happening in different places around the world, probably including Canada to a certain extent, where people are deciding to abort. They say, well, we've got enough girls, we want boys. So this has been a controversial thing, and selectively aborting fetuses that will grow up into people who have Down syndrome. That is something else that people do. So think about it. This is a very controversial thing, but it's something that's taking place right now. Okay, so another thing that's happening is that there are kind of comical attempts at eugenics by people who are trying to increase desirable characteristics in the population. And so there are a couple of weird people who've done things like create sperm banks where, a sperm bank is where women can go to get a sperm sample to fertilize her eggs using in vitro fertilization. If she can't have children, if it's a married couple and the husband is sterile for some reason, they could go to a sperm bank to get sperm. There are other reasons why people might want to do that, lots of other reasons. But there are some nutty people around, one of which I'll tell you about in a minute, who created a sperm bank that was only for the sperm of men who had won the Nobel Prize. And his reasoning behind that was that those men are smarter than regular men, and so you can go to the sperm bank, and if you get sperm from a Nobel Prize laureate, then you'll have a smarter child. I don't think that's actually been proven, or I don't think that's borne out to be true, but that is the attitude. This is the guy here. His name is Robert K. Graham. And he started a sperm bank that contains sperm from only Nobel Prize winners, right? So he was a multimillionaire because he invented plastic eyeglasses. He was an optometrist who invented plastic eyeglasses. So if you're wearing plastic eyeglasses right now like I am, you've probably contributed some money to this man's estate, this crazy man's estate who started up the Nobel Prize winning sperm I I've only known one person in my life who won the Nobel Prize, Michael Smith, who won the Nobel Prize. I worked in his lab in 1993 when he won the Nobel Prize, and I don't know if, I don't know if he was ever donating any sperm to sperm banks, but I doubt it. Anyway, there are other people who go to the sperm bank, and one of the things, people, couples who can't have their own kids, or they go to the sperm bank, and one of the first questions they ask apparently is, was the man who donated this sperm, was he good looking? Because we want our child to be good looking so that they'll have a better deal in life. So these attitudes do exist, and that is a form of eugenics. Okay, here's an interesting character. This was Dr. Cecil Jacobson, just for, I guess I put him here for amusement, if nothing else. But he got into trouble because he was a fertility doctor who did in vitro fertilizations on hundreds of women. And in those in vitro fertilizations, he was supposed to be mixing together sperm. He was supposed to be mixing a woman's eggs together with sperm from an anonymous donor or from her husband or something like that. And in reality, he snuck in his own sperm. He put his own sperm in instead. And so he ended up being the father of 250 children or something like that. Little example of reproductive technology gone mad. He is mad if he did that. I mean, why else would you do something like that? Why would you want to father 250 children who didn't know that you were their father? That makes no sense unless you're a little bit kooky. All right, so this has been quite a long lecture. I apologize, but we got through the human reproductive system. So we talked about the advantage. We started by talking about the advantages of sexual reproduction to evolution. And then we talked about forms of sexual versus asexual reproduction, gametogenesis, which is how we create sperms and eggs. We talked about the hormonal control of ovulation, making sure that only one egg is available to be fertilized every month. We talked about the specifics of the male and female reproductive anatomy, and we talked about some things that can go wrong with the male and female reproductive anatomy. In the case of men, that would be erectile dysfunction and enlarged prostate. In women, that would be endometriosis and ectopic pregnancies. We talked about contraception and sexually transmitted diseases. We also talked about what happens after a woman becomes pregnant, the gestation period, the 3 trimesters, and embryogenesis taking place in the first 2 months basically. Organogenesis. Organogenesis taking place in the first 2 months. We talked about determination of sex using chromosomes and how in humans and most mammals, the presence of the Y chromosome is what determines whether you're male. If you don't have the Y chromosome, you become a female by default, as is the case with Turner syndrome, for example. And then we talked about birth defects, and then we finished by talking about the philosophical questions surrounding eugenics. Okay, good luck on the quiz on human reproduction, and there'll probably be a lot of questions about reproduction on the final, so good luck.